Right now, as you settle into this quiet moment, millions of invisible partners are working to keep you alive. The air you breathe carries oxygen crafted by ancient cyanobacteria and towering trees. Your body hosts beneficial microbes whose ancestors helped shape the very atmosphere. Every cell in your being contains mitochondria, once free living bacteria that chose cooperation over competition billions of years ago. You are not separate from nature. You are woven into its intricate tapestry, sustained by a web of relationships spanning every corner of our planet. This is the story of biodiversity, the magnificent variety of life that makes existence possible. The word biodiversity rolls off the tongue easily enough, yet beneath its simple syllables lies one of the most complex and beautiful concepts in all of science. At its heart, biodiversity is the variety of life on Earth, but this definition barely scratches the surface of what makes our planet so remarkably alive. Like a Russian nesting doll, biodiversity reveals layer upon layer of complexity, each level containing worlds within worlds, each essential for the functioning of the whole. To truly understand biodiversity, we must first recognize that it operates on three distinct but interconnected levels. Think of these as three different lenses through which we can view the richness of life, each revealing patterns and processes invisible to the others. Together, they form a complete picture of how life maintains itself across time and space. The first level, genetic diversity, exists within the very building blocks of life itself. Every individual organism, from the smallest bacterium to the largest blue whale, carries within its cells a unique genetic library written in the four-letter alphabet of DNA. This genetic variation is the raw material of evolution, the foundation upon which all other forms of diversity rest. Consider the humble domestic dog, descended from wolves, yet now existing in hundreds of breeds, each shaped by selective breeding that highlighted different aspects of the underlying genetic variation. A Chihuahua and a Great Dane share the vast majority of their DNA, yet the expression of that genetic information has been molded into forms so different they barely seem related. In wild populations, genetic diversity serves as a kind of insurance policy against an uncertain future. A forest of genetically identical trees might seem pristine and orderly, but it represents a dangerous gamble. Should a new disease emerge or environmental conditions shift dramatically, the entire population could be vulnerable. In contrast, a genetically diverse forest contains individuals with slightly different responses to stress, disease resistance, and environmental tolerance. Some trees might succumb to a pathogen, but others will survive, carrying their resistant genes forward to the next generation. This genetic insurance becomes particularly crucial when we consider the whitebark pine forests of North America. These high-elevation trees face mounting pressure from an introduced fungus called white pine blister rust. In genetically uniform stands, the disease spreads like wildfire, leaving vast expanses of dead trees. But in areas where genetic diversity remains high, researchers have found individual trees with natural resistance to the fungus. These survivors become the foundation for restoration efforts, their unique genetic combinations offering hope for the species' future. The second level of biodiversity, species diversity, is perhaps the most familiar to us. This is the variety we see when we walk through a forest and notice different types of trees, or when we peer into tide pools and marvel at the array of creatures clinging to the rocks. Species diversity encompasses not just the number of different species in a given area, but also their relative abundance and the roles they play in their ecosystem. A meadow dominated by a single type of grass represents low species diversity, even if millions of individual plants carpet the landscape. In contrast, a prairie containing dozens of different plant species, even if some are quite rare, represents much higher diversity. But the story becomes more nuanced when we consider that the rare species might play critical roles disproportionate to their numbers. The few remaining wild lupines in a degraded prairie might be the only host plants for endangered Carner blue butterflies, making their preservation essential for maintaining the ecosystem's full complement of species. Species diversity varies dramatically across the planet, following patterns that have fascinated scientists for centuries. Travel from the poles toward the equator, and you'll generally encounter an increasing array of species. A single tree in the Amazon rainforest might host more species of ants than exist in all of Alaska. This latitudinal gradient in diversity reflects the interaction of climate, evolutionary history, and ecological opportunity. Near the equator, stable warm temperatures and abundant rainfall create conditions where specialization can flourish. Species can afford to develop narrow ecological niches because resources are predictable and competition drives ever finer divisions of the available opportunities. The third level, ecosystem diversity, encompasses the variety of habitats, communities, and ecological processes across landscapes and regions. From the perspective of a migrating bird, the Earth presents a patchwork of different ecosystem types, each offering distinct resources and challenges. Wetlands provide stopover sites rich in invertebrate food. Forests offer nesting habitat and protection from predators. Grasslands yield seeds and small mammals. The diversity of these ecosystem types across the landscape creates the spatial mosaic that makes complex life cycles possible. Ecosystem diversity operates at multiple scales, from the microhabitats within a single forest to the continental scale distribution of biomes. A fallen log in an old-growth forest represents its own ecosystem, supporting specialized fungi, insects, and salamanders found nowhere else in the forest. At a larger scale, the patchwork of meadows, streams, and forest stands across a watershed, creates the ecosystem diversity that supports species with different habitat requirements throughout their life cycles. These three levels of biodiversity are not independent, but deeply interconnected, each influencing and supporting the others in complex feedback loops. Genetic diversity within species provides the raw material for adaptation to different environments, potentially leading to the formation of new species over time. The presence of multiple species creates the ecological interactions that shape ecosystem structure and function. Different ecosystem types, in turn, provide the environmental variation that drives genetic and species diversification. Consider how these levels interact in a coral reef ecosystem. The genetic diversity within coral species allows some individuals to tolerate slightly higher temperatures or different light conditions, creating microhabitat variation across the reef. This environmental heterogeneity supports different assemblages of fish, invertebrates, and algae, increasing species diversity. The activities of these diverse species, from the grazing of parrotfish to the cleaning services of wrasses, help maintain the ecosystem's structure and function. When this system is functioning well, it creates conditions that support continued evolution and diversification. The loss of diversity at any level can cascade through the others, creating feedback loops that accelerate decline. When genetic diversity within a species becomes too low, the species becomes vulnerable to extinction. The loss of species reduces the ecological interactions that maintain ecosystem structure. Simplified ecosystems provide fewer opportunities for specialization and adaptation, reducing the generation of new genetic and species diversity. This interconnectedness means that biodiversity conservation must address all three levels simultaneously. Protecting a single charismatic species is not enough if the genetic diversity within that species is critically low. Preserving large numbers of species means little if they are confined to simplified habitats that cannot support their complex life cycles. Maintaining ecosystem diversity requires attention to the genetic health of the species that create and maintain those ecosystems. As we explore deeper into the world of biodiversity, we begin to see that these patterns of diversity are not randomly distributed across our planet. Instead, they follow predictable rules shaped by climate, geography, and evolutionary history. Some places on Earth concentrate extraordinary amounts of biodiversity into relatively small areas, becoming treasure troves of biological wealth that support far more than their fair share of global diversity. Understanding where these patterns emerge and why they persist will guide us toward appreciating how geography and environment shape the distribution of life across our remarkable planet. If you could see the Earth from space with eyes that revealed biodiversity instead of geography, our planet would appear as a magnificent tapestry of light and shadow. Brilliant bands of intensity would wrap around the equator, glowing with the concentrated richness of tropical rainforests and coral reefs. These luminous belts would gradually fade toward the poles, where life persists in more subdued but no less remarkable forms. Islands would sparkle like scattered jewels, each harboring unique evolutionary experiments. Mountain ranges would display vertical rainbows of diversity, their slopes painted in bands that mirror the journey from equator to pole. This imaginary view would reveal one of ecology's most fundamental patterns. Biodiversity is not evenly distributed across our planet. Instead, it follows predictable rules that have shaped the distribution of life for millions of years. Understanding these patterns helps us appreciate why some places teem with countless species, while others support only a few hardy specialists, and why the protection of certain regions matters so much for global conservation. The most striking pattern is the latitudinal gradient of diversity, a phenomenon so consistent that it ranks among ecology's few truly universal rules. As you travel from the poles toward the equator, the number of species in most groups of organisms increases dramatically. A forest in Costa Rica might contain more tree species in a single hectare than exist in all of Canada. The bird life of a small reserve in Ecuador can exceed the diversity found across entire continents in the far north. This gradient appears in nearly every group of organisms, from bacteria to mammals, from flowering plants to marine fish. Several factors combine to create this remarkable pattern. Temperature plays a crucial role, as warm conditions generally support higher metabolic rates and faster generation times, accelerating the pace of evolution and speciation. In the tropics, the absence of harsh winters means that organisms can specialize in narrower ecological niches without facing the periodic catastrophes that force temperate species to be generalists. A tropical insect might evolve to feed on a single species of plant, timing its life cycle to coincide perfectly with the plant's fruiting schedule. Such specialization would be impossible in environments where winter forces all activity to cease for months at a time. The stability of tropical climates over geological time has also allowed diversity to accumulate without the periodic extinctions that have repeatedly reset diversity in temperate regions. While northern forests have been repeatedly scoured by ice ages, tropical regions have served as refugia where species could persist and continue evolving. This head start, combined with the ongoing rapid pace of speciation in warm climates, has created the extraordinary diversity we see in tropical regions today. Energy availability also contributes to the latitudinal gradient. Near the equator, intense solar radiation and abundant rainfall support high primary productivity, creating the energetic foundation for complex food webs. More energy flowing through an ecosystem can support more species, each capturing and utilizing different fractions of the total energy available. In contrast, the limited growing seasons and lower productivity of polar regions can support only simpler food webs with fewer species. But the story of global diversity patterns extends far beyond simple latitudinal gradients. Elevation creates its own diversity patterns, often compressed into remarkably small distances. A climb up a tall mountain can take you through as many different life zones as a journey from temperate regions to the tropics. At the base of the Andes in Ecuador, you might begin in lowland rainforest rich with jaguars, toucans, and thousands of insect species. As you ascend, cloud forests emerge, shrouded in mist and dominated by epiphytes that capture moisture from the air. Higher still, Paramo grasslands stretch across windswept plateaus, home to specialized plants and animals found nowhere else on Earth. Finally, snow-capped peaks support only the hardiest alpine specialists. This elevational diversity reflects the same factors that drive latitudinal patterns, but compressed into vertical space. Temperature decreases with elevation, just as it does with latitude. Precipitation patterns change, often creating distinct wet and dry zones at different elevations. The result is a series of stacked ecosystems, each with its own characteristic species and ecological processes. Islands present some of the most fascinating patterns in global biodiversity. Despite their relatively small size, islands often harbor extraordinary numbers of endemic species found nowhere else on Earth. The Galapagos Islands, covering less area than Connecticut, contain species that helped inspire Darwin's theory of evolution. Madagascar, separated from Africa over 100 million years ago, has evolved a flora and fauna so distinctive that it's often called the eighth continent. Nearly 90% of its plant species and 80% of its animal species are endemic. Island biogeography theory helps explain these patterns. When ancestral species first arrive on isolated islands, they encounter environments with fewer competing species and often vacant ecological niches. Over time, populations adapt to local conditions and may split into multiple species, each specialized for different aspects of the island environment. The famous Darwin's finches of the Galapagos illustrate this process beautifully. From a single ancestral species, at least 18 different finch species have evolved, each with beak shapes adapted for different food sources, from large seeds to flower nectar to blood. The size of islands influences their diversity in predictable ways. Larger islands can support more species because they contain more habitats and can sustain larger populations that are less prone to extinction. The distance from mainland source populations also matters. Islands far from potential colonists will have fewer species simply because fewer organisms manage to make the journey. These patterns hold true whether we're talking about actual islands surrounded by water or habitat islands, like isolated mountaintops surrounded by unsuitable lowlands. Biodiversity hotspots represent the planet's most spectacular concentrations of species richness and endemism. These 36 regions, covering just 2.4% of Earth's land surface, support over half of all plant species and 43% of vertebrate species as endemics. The criteria for hotspot designation are stringent. A region must contain at least 1,500 endemic plant species and have lost at least 70% of its original habitat. The Mediterranean basin exemplifies a biodiversity hotspot. This region, stretching from Portugal to Jordan and from Italy to Morocco, contains over 25,000 plant species, more than half of which are endemic. The complex geography of peninsulas, islands, and mountain ranges has created countless isolated populations that have evolved into distinct species. The climate, with its distinctive pattern of wet winters and dry summers, has selected for unique adaptations found in few other places on Earth. Other hotspots tell similar stories of geographic complexity, climatic distinctiveness, and evolutionary isolation. The California floristic province owes its extraordinary plant diversity to its Mediterranean climate, complex topography, and position at the intersection of different biogeographic regions. The Cape Floral region of South Africa, smaller than Portugal, contains more plant species than the entire British Isles. The Western Ghats of India, a mountain range running along the country's western coast, harbors thousands of endemic species in forests isolated by surrounding dry lands. Marine diversity patterns follow some similar rules, but with important differences. Coral reef regions, particularly the Coral Triangle of Southeast Asia, represent the marine equivalent of tropical rainforests. This region, encompassing parts of Indonesia, Malaysia, the Philippines, and Papua New Guinea, contains about 30% of the world's coral reefs and supports over 3,000 species of fish. Like terrestrial hotspots, this marine diversity reflects geographic complexity, climatic stability, and evolutionary opportunity. Ocean currents create their own biogeographic patterns, transporting larvae and nutrients across vast distances while creating barriers to dispersal in other directions. The Gulf Stream carries warm water and tropical species far north along the eastern coast of North America, while the cold California current brings northern species south along the Pacific coast. These currents help explain why the marine life of Florida's Atlantic coast differs dramatically from that of its Gulf Coast, despite their geographic proximity. Freshwater systems add another layer to global diversity patterns. Rivers, lakes, and wetlands often harbor high levels of endemism because freshwater organisms cannot easily disperse across terrestrial barriers. The ancient lakes of the world, such as Lake Baikal in Siberia and Lake Tanganyika in Africa, have become evolutionary laboratories where endemic species have proliferated over millions of years. Lake Baikal alone contains about 1,700 endemic species, including the world's only freshwater seal. These patterns of diversity distribution have profound implications for conservation. The concentration of species in hotspots means that protecting relatively small areas can preserve disproportionate amounts of global diversity. However, this also means that these regions face enormous pressure as they often coincide with areas of high human population density and agricultural productivity. Understanding where diversity occurs, why it concentrates in particular places, and how it responds to environmental change becomes crucial for maintaining the biological wealth that supports all life on Earth. As we contemplate these global patterns, we begin to see that biodiversity is not just a collection of individual species scattered randomly across the landscape. Instead, it represents the accumulated result of millions of years of evolution shaped by climate, geography, and ecological opportunity into predictable patterns that repeat across the globe. These patterns provide the foundation for understanding how diversity functions within ecosystems, creating the complex structures and processes that we will explore as we delve deeper into the architecture of life. In the same way that architects design buildings with carefully planned spaces, levels, and support structures, biodiversity constructs ecosystems with intricate three-dimensional arrangements that determine where life can flourish. But unlike human architecture, where designers impose their vision on raw materials, ecological architecture emerges from the interactions of countless organisms, each pursuing its own survival while collectively creating the complex structures that define natural communities. Stand in an old growth forest and look up. What you see is not simply a collection of trees, but a carefully orchestrated vertical arrangement of life zones, each supporting its own community of specialists. The forest floor, dim and rich with decomposing organic matter, hosts salamanders, millipedes, and countless fungi. Above, the understory creates a filtered light environment where shade-tolerant shrubs and herbs have evolved broad leaves to capture every photon. Higher still, the main canopy forms a continuous roof of interwoven branches, creating an aerial habitat so distinct from the ground below that some animals spend their entire lives without ever touching the earth. And finally, emergent giants pierce through this canopy roof, their crowns bathed in full sunlight and buffeted by winds unknown in the sheltered world below. This vertical stratification represents one of the most fundamental ways that biodiversity creates ecosystem structure. Each layer exists because different species have evolved to exploit different portions of the available space and resources. The result is a three-dimensional mosaic that dramatically increases the total amount of living space available within a given area. A forest with this complex structure can support far more species than a simple grassland of the same area, precisely because it provides so many more ecological niches. The creation of this architecture begins with the foundation species, those organisms whose physical presence creates the basic structure that others depend upon. In forests, trees serve this role not just through their obvious contribution of vertical structure, but through their more subtle influences on light, temperature, humidity, and soil chemistry. A large tree doesn't simply occupy space, it modifies the environment around it, creating gradients of conditions that other species can exploit. Consider how a single large oak tree transforms its surroundings. Its canopy intercepts rainfall, creating a drier zone directly beneath its crown while funneling water to a wetter zone around its drip line. Its roots extend far beyond the reach of its branches, creating underground networks that modify soil chemistry and provide pathways for mycorrhizal fungi. As it drops leaves each autumn, it creates an ever-changing patch of rich organic matter that supports different communities of decomposers than the surrounding forest floor. The bark provides attachment sites for lichens and mosses, while cavities in its trunk offer nesting sites for birds and mammals. Through these multiple modifications of its environment, a single tree creates dozens of microhabitats within its sphere of influence. Aquatic ecosystems demonstrate equally sophisticated architectural principles. though the medium of water creates different opportunities and constraints. Coral reefs represent perhaps the most spectacular example of biodiversity generated architecture in marine environments. The calcium carbonate skeletons of countless coral polyps, accumulated over centuries, create massive three-dimensional structures that rival any terrestrial habitat in their complexity. A healthy coral reef displays the same kind of vertical zonation seen in forests, but with unique adaptations to the aquatic environment. Different coral species dominate at different depths, each adapted to particular light levels and wave energies. Massive boulder corals form the foundation in deeper waters, their robust skeletons capable of withstanding strong currents. Branching corals create complex three-dimensional tangles in shallower areas, providing hiding places for juvenile fish and small invertebrates. Plate corals extend horizontal surfaces to capture maximum light in the filtered underwater environment. But the architectural complexity of reefs extends far beyond the corals themselves. Countless other organisms contribute to and modify the reef structure. Boring sponges and mollusks create cavities within the coral framework, while coralline algae cement loose fragments together. The result is a porous, cave-riddled structure that provides an enormous variety of spaces for different organisms to inhabit. The cryptic fauna hidden within reef frameworks often includes more species than the visible fish and corals swimming around the reef's exterior. In freshwater systems, architecture emerges through different mechanisms but follows similar principles. Submerged and emergent vegetation creates the structural foundation for lake and pond communities. Cattails and bulrushes rising above the water surface provide nesting sites for birds and attachment points for climbing insects. Their submerged stems offer protection for young fish and surfaces for algae and aquatic invertebrates. Floating-leaved plants like water lilies create a two-dimensional habitat at the water's surface, providing basking sites for turtles and hunting platforms for herons, while their underwater roots anchor in the sediment below. The three-dimensional nature of aquatic plant communities becomes particularly apparent in spring, when many temperate lakes and ponds experience dramatic seasonal changes in structure. As water temperatures rise, submerged plants begin growing from the bottom up, creating an underwater forest that transforms the habitat available to fish and aquatic invertebrates. This seasonal architecture influences breeding patterns, migration routes, and feeding strategies throughout the aquatic community. Even seemingly simple ecosystems reveal unexpected architectural complexity when examined closely. Grasslands, often perceived as two-dimensional carpets of vegetation, actually display sophisticated vertical organization. Prairie grasses with different heights and growth forms create multiple canopy layers, each intercepting light and precipitation differently. Deep-rooted species like big bluestem send shoots far into the soil, while shallow-rooted species spread horizontally near the surface. The result is a complex, three-dimensional root architecture below ground that mirrors the above-ground structural diversity. The architectural role of biodiversity becomes perhaps most apparent when key structural species are removed from ecosystems. The loss of large trees in a forest doesn't simply reduce the number of tree species, it collapses entire vertical zones of habitat. Canopy-dwelling species suddenly have no place to live, while shade-adapted understory species face lethal exposure to direct sunlight. The complex microclimates created by tree canopies disappear, replaced by simpler, more uniform conditions that support fewer species overall. Similar architectural collapses occur in aquatic systems, when key structural species are lost. The removal of kelp forests along temperate coastlines eliminates not just the kelp itself, but the entire three-dimensional habitat it created. Fish species that depend on the vertical complexity of kelp forests for feeding and protection find themselves in barren underwater deserts. Sea urchins, formerly controlled by predators that sheltered in the kelp canopy, can overgraze the remaining algae and prevent forest recovery. The creation of ecosystem architecture through biodiversity involves positive feedback loops that tend to increase complexity over time. As structural species modify their environment, they create new opportunities for other species to colonize and establish. These new arrivals often contribute their own modifications, further increasing the habitat's complexity and its capacity to support even more species. Over ecological and evolutionary time, this process can lead to the extraordinary architectural complexity we see in mature ecosystems. Succession provides a clear window into this architectural development process. Early successional communities typically have simple, largely two-dimensional structure dominated by fast-growing herbaceous plants. As succession proceeds, shrubs and eventually trees add vertical complexity, creating the multi-layered structure characteristic of mature forests. Each stage of succession supports different communities of animals, with overall diversity generally increasing as architectural complexity develops. Human activities often simplify ecosystem architecture, reducing the three-dimensional complexity that supports diverse communities. Agricultural monocultures represent extreme architectural simplification, replacing the complex vertical and horizontal structure of natural plant communities with uniform stands of single crop species. Even forest management practices that favor single-aged, single-species stands dramatically reduce the architectural complexity compared to natural forests with their mix of species, ages, and structural forms. Understanding ecosystem architecture helps explain why some conservation strategies focus on protecting structural diversity rather than just species diversity. Maintaining old-growth forests preserves not just the tree species themselves, but the complex three-dimensional habitats they create. Protecting coral reefs safeguards not only the coral species, but the architectural framework that supports entire marine communities. The architectural complexity created by biodiversity also influences ecosystem functioning in ways that extend far beyond simply providing habitat space. The three-dimensional structure affects how energy and nutrients flow through ecosystems, how water and air move through different layers, and how organisms interact with each other and their physical environment. As we explore deeper into the functional roles that different species play in maintaining ecosystem processes, we'll see how the structural foundation created by biodiversity supports the complex web of activities that keep ecosystems running smoothly. Within the complex architecture of ecosystems, every species performs specific roles that contribute to the functioning of the whole. Like workers in a vast, self-organizing city, different organisms specialize in particular tasks that keep essential processes running smoothly. Some capture energy from the sun, others break down waste, still others transport materials from place to place. This division of labor, known as functional diversity, transforms collections of individual species into integrated systems capable of maintaining themselves over time. The concept of functional groups helps us understand how this biological workforce operates. Rather than focusing on taxonomic relationships, which species are most closely related to each other, functional classification groups, organisms based on what they do in their ecosystems. A hummingbird and a butterfly might be separated by hundreds of millions of years of evolutionary history, yet both function as pollinators, transferring genetic material between flowering plants. A vulture and a beetle occupy completely different branches of the tree of life, but both serve as decomposers, breaking down dead organic matter and returning nutrients to the ecosystem. At the foundation of virtually all terrestrial and shallow aquatic ecosystems stand the primary producers, organisms capable of capturing energy directly from the sun and transforming it into the chemical bonds of organic molecules. Green plants dominate this role in most familiar ecosystems, but they share the stage with algae, cyanobacteria, and other photosynthetic organisms. These primary producers don't simply feed themselves, they create the energetic foundation that supports all other life in their communities. The efficiency and diversity of primary production varies dramatically among ecosystems, creating cascading effects throughout entire food webs. In tropical rainforests, dozens of tree species with different leaf shapes, heights, and light requirements create a complex mosaic of photosynthetic activity. Some species specialize in capturing the brightest light at the top of the canopy, while others excel at photosynthesis in the dim conditions of the forest floor. This functional diversity among producers maximizes the total amount of solar energy captured and converted into biological energy, supporting the extraordinary diversity of consumers that tropical forests can sustain. Primary consumers form the next functional layer, converting plant material into animal biomass. But even within this seemingly straightforward role, remarkable functional diversity emerges. Grazing animals like zebras and wildebeest specialize in eating grasses, maintaining savanna ecosystems through their feeding activities. Browsing animals such as giraffes and elephants focus on woody plants, shaping forest structure through their selective feeding. Frugivorous animals, like many primates and birds, consume fruits, inadvertently dispersing seeds and maintaining plant genetic diversity across landscapes. The timing and selectivity of different herbivores creates complex patterns of plant community structure. Some herbivores feed continuously throughout the year, maintaining steady pressure on plant populations. Others concentrate their feeding during specific seasons, allowing plants to recover and reproduce during herbivore-free periods. Specialist herbivores that feed on only one or a few plant species can become tightly linked to their food sources, while generalist feeders can switch among different plants as availability changes. Secondary consumers occupy the role of predators, controlling herbivore populations and indirectly influencing plant communities through these top-down effects. But predation itself encompasses remarkable functional diversity. Ambush predators like praying mantises and some snakes wait motionlessly for prey to approach, while pursuit predators like cheetahs and wolves actively chase down their victims. Some predators specialize in particular types of prey, while others opportunistically consume whatever becomes available. The functional role of predators extends far beyond simply removing individuals from prey populations. Different predation strategies create different selective pressures on prey species, driving the evolution of various anti-predator adaptations. The presence of predators also influences prey behavior, affecting where and when herbivores feed, and thus indirectly shaping plant community structure. Wolves in Yellowstone National Park don't just kill elk. Their presence changes elk behavior, reducing browsing pressure on willows and aspens in areas where elk feel vulnerable to predation. Decomposers perform one of the most essential, yet often overlooked functions in ecosystems. Without the constant work of bacteria, fungi, and countless invertebrates breaking down dead organic matter, nutrients would remain locked in dead tissues and unavailable to living organisms. The functional diversity within decomposer communities ensures that different types of organic matter are efficiently processed and recycled. Fungi excel at breaking down tough plant materials like cellulose and lignin, often serving as the primary decomposers of woody material. Bacteria dominate the decomposition of softer tissues and are particularly important in aquatic environments. Invertebrate decomposers like millipedes, springtails, and earthworms physically fragment dead material, increasing its surface area and making it more accessible to microbial decomposers. Each group contributes different enzymes and processing capabilities, working together to completely recycle the complex molecules that make up living tissues. The rate and completeness of decomposition varies dramatically among ecosystems, largely reflecting the functional diversity of decomposer communities. In tropical rainforests, where warm temperatures and high humidity support diverse and active decomposer communities, dead leaves may disappear within weeks of hitting the forest floor. In contrast, the limited decomposer diversity in Arctic tundra means that organic matter can persist for decades or centuries, creating vast stores of carbon in frozen soils. Pollinators represent another crucial functional group, one whose importance becomes apparent whenever they decline or disappear. These organisms transport genetic material between individual plants, enabling sexual reproduction and maintaining genetic diversity within plant populations. The functional diversity of pollinator communities ensures that different plant species receive appropriate pollination services throughout the growing season. Different pollinators possess different body sizes, flower preferences, and flight patterns, making them effective at pollinating different types of plants. Large bees can force their way into snapdragons and other flowers with restrictive architectures, while tiny flies can access minute flowers that larger pollinators ignore. Long-tongued moths and butterflies specialize in flowers with deep nectar tubes, while short-tongued bees focus on more accessible blooms. Some pollinators are active only during specific times of day or seasons, providing pollination services to plants that bloom during those periods. The decline of pollinator diversity can have cascading effects throughout plant communities. When specialist pollinators disappear, their associated plant species may suffer reduced reproduction, leading to population declines that affect the herbivores and predators that depend on those plants. The loss of pollinator functional diversity can simplify plant communities, reducing the resources available to other functional groups and ultimately decreasing overall ecosystem complexity. Seed dispersers form yet another critical functional group, responsible for moving plant propagules away from parent plants and into suitable germination sites. Different dispersal agents move seeds different distances and deposit them in different microenvironments, influencing plant population structure and genetic diversity. Large mammals like elephants and tapirs can transport seeds across vast distances, maintaining genetic connectivity between distant plant populations. Birds often deposit seeds in elevated perches where they defecate, creating favorable germination conditions. Small mammals may bury seeds in scattered caches, providing both dispersal and protection from seed predators. The functional diversity of seed dispersers becomes particularly important in fragmented landscapes, where long-distance dispersal may be essential for maintaining plant populations in isolated habitat patches. The loss of large frugivorous animals can severely limit the dispersal of large seeded plants, leading to recruitment failures and eventual population declines. Ecosystem engineers represent a special category of functional group whose activities modify the physical environment in ways that affect many other species. Beavers exemplify this role, transforming flowing streams into complex wetland systems through their dam building activities. Their engineering creates habitat for countless other species while dramatically altering hydrology across entire watersheds. Earthworms serve as ecosystem engineers in many soils, mixing organic and mineral layers while creating channels that improve water infiltration and root penetration. The functional diversity approach reveals that ecosystem stability depends not just on the number of species present, but on maintaining the full range of functional roles that keep ecosystems operating. Functional redundancy, having multiple species that perform similar roles, provides insurance against the loss of any single species. But functional complementarity, having species that perform different aspects of important processes, ensures that all necessary functions are adequately performed. As we explore the specific services that these functional groups provide to both natural ecosystems and human societies, we begin to see how the intricate division of labor among species creates the life support systems that sustain our planet's remarkable diversity of life. Every morning, as sunlight filters through your window and you take your first conscious breath of the day, you participate in an ancient exchange that connects you to every corner of the living world. The oxygen filling your lungs was produced by photosynthetic organisms scattered across continents and oceans. The water you drink has been purified by wetlands, filtered through forest soils, and recharged in aquifers maintained by plant roots. The stable climate that makes your existence possible emerges from the collective activities of countless species regulating atmospheric chemistry, reflecting solar radiation, and moderating temperature extremes. These benefits that flow from healthy ecosystems to support all life, including human societies, are called ecosystem services. They represent the practical manifestation of biodiversity's functional roles, translating the abstract concept of ecological processes into concrete benefits that sustain life on Earth. Understanding these services reveals how deeply human welfare depends on maintaining the complex biological systems that have evolved over millions of years. Climate regulation stands among the most fundamental and globally significant ecosystem services. The world's forests, grasslands, and oceans function as vast climate control systems, moderating temperature, humidity, and atmospheric composition on both local and global scales. Trees and other vegetation absorb carbon dioxide during photosynthesis, removing this greenhouse gas from the atmosphere and storing it in wood, leaves, and soil. A single mature tree can absorb 48 pounds of carbon dioxide annually, while an acre of forest may store thousands of pounds of carbon in above and below ground biomass. But climate regulation extends far beyond carbon storage. Forests influence local and regional weather patterns through evapotranspiration, the process by which plants release water vapor into the atmosphere. A large tree can transpire hundreds of gallons of water per day, cooling the surrounding air and increasing local humidity. This process helps moderate temperature extremes and can influence precipitation patterns downwind. The Amazon rainforest generates much of its own rainfall through this process, creating a self-sustaining hydrological cycle that maintains forest conditions across an area larger than the continental United States. Urban forests provide localized climate services that directly improve human comfort and reduce energy demands. Tree-lined streets can be 10 to 15 degrees cooler than nearby areas without vegetation. The shade and evapotranspiration from mature street trees can reduce summer air conditioning costs by 30% or more, while strategically placed trees can reduce winter heating costs by blocking cold winds. Cities with extensive tree cover experience less severe heat island effects, making them more livable during extreme weather events. Water purification and regulation represent another critical category of ecosystem services. Wetlands function as nature's kidneys, filtering pollutants from water as it flows through their complex plant and soil systems. The roots of wetland plants absorb excess nutrients like nitrogen and phosphorus that might otherwise cause harmful algal blooms in downstream lakes and rivers. Wetland soils trap sediments and bind heavy metals, preventing these contaminants from reaching drinking water supplies. Forests provide equally important water services through their influence on watershed hydrology. Tree canopies intercept rainfall, reducing erosion by breaking the force of falling raindrops. Forest soils, enriched with organic matter from decomposing leaves and roots, act like giant sponges, absorbing rainfall and releasing it gradually to streams and groundwater. This natural water storage system helps prevent both floods during heavy rains and droughts during dry periods. The economic value of these water services becomes apparent when they're lost or degraded. New York City's water supply system illustrates this principle beautifully. Rather than building expensive water treatment facilities, the city invested in protecting the watersheds that naturally purify water flowing to its reservoirs. This ecosystem-based approach provides clean water to 9 million people at a fraction of the cost of technological alternatives. Soil formation and maintenance represent ecosystem services that operate over longer timescales but remain equally essential for life on Earth. The thin layer of soil that covers much of the planet's land surface represents millions of years of accumulated biological activity. Countless generations of plants, animals, fungi, and microorganisms have lived, died, and decomposed to create the complex mixture of organic matter, minerals, and living organisms that make up healthy soils. Different organisms contribute specialized services to soil development and maintenance. Plant roots create channels that improve water infiltration and provide pathways for soil organisms. Mycorrhizal fungi extend root systems and help plants access nutrients from greater volumes of soil. Earthworms and other invertebrates mix organic and mineral soil layers while creating stable soil aggregates that resist erosion. Bacteria and other microorganisms decompose organic matter and make nutrients available to plants. The diversity of soil organisms directly influences soil health and productivity. Soils with high biological diversity tend to be more fertile, better able to retain water and nutrients, and more resistant to erosion and disease. Agricultural systems that maintain soil biological diversity through practices like cover cropping and reduced tillage often achieve higher yields with fewer external inputs than simplified systems that rely heavily on synthetic fertilizers, and pesticides. Pollination services provide a direct link between biodiversity and human food security. Approximately 1/3 of the food crops that humans consume depend on animal pollination, including many fruits, vegetables, nuts, and seeds that provide essential nutrients for human health. The economic value of pollination services globally exceeds $200 billion annually, yet this service is provided free by wild and managed pollinators. The diversity of pollinator communities ensures reliable pollination across different crops and growing seasons. Native bees often prove more effective than honeybees at pollinating certain crops, while different pollinator species remain active under different weather conditions. This functional diversity provides insurance against pollination failures that could result from the decline of any single pollinator species. Pest control represents another economically significant ecosystem service provided by biodiversity. Natural enemies of crop pests, including predatory insects, parasitic wasps, spiders, and birds, can substantially reduce pest populations without the environmental costs associated with pesticide use. A diverse community of natural enemies provides more effective and stable pest control than reliance on a few dominant species. Birds alone provide billions of dollars worth of pest control services annually. A single barn owl can consume over 1,000 rodents per year, while insectivorous birds in forests can remove enough caterpillars and other herbivorous insects to measurably increase tree growth and survival. Coffee plantations with higher bird diversity typically experience less damage from the coffee berry borer and other insect pests. Disease regulation emerges from the complex interactions among hosts, pathogens, and the broader biological community. Biodiversity can reduce disease transmission through several mechanisms. The dilution effect occurs when diverse host communities include many species that cannot support particular pathogens, reducing the probability that susceptible hosts will encounter infected individuals. Some non-host species may actively remove pathogens from the environment or support populations of organisms that compete with or consume pathogens. The emergence of Lyme disease in North America illustrates how biodiversity loss can increase disease risk. In diverse forest communities, many small mammals serve as hosts for tick larvae, but not all of these hosts can support the bacterium that causes Lyme disease. As forest fragmentation reduces mammal diversity, white-footed mice, which are excellent reservoirs for Lyme disease bacteria, become more dominant. This increases the proportion of infected ticks and raises the risk of human disease transmission. Genetic resources represent a category of ecosystem services that provides the raw material for developing new medicines, crops, and other useful products. Wild relatives of crop plants contain genetic variations that can be bred into cultivated varieties to improve disease resistance, nutritional content, or adaptation to changing environmental conditions. Many modern medicines derive from compounds originally discovered in wild plants, animals, or microorganisms. The rosy periwinkle from Madagascar exemplifies the potential value of genetic resources. Compounds from this plant are used to treat childhood leukemia and Hodgkin's disease, generating hundreds of millions of dollars in pharmaceutical sales annually. Yet Madagascar has lost over 80% of its original vegetation, and countless species with potential medical applications have likely been lost forever. Cultural and spiritual services provided by biodiversity are harder to quantify economically, but remain essential for human well-being. Natural areas provide opportunities for recreation, education, and spiritual renewal that contribute to both physical and mental health. Many indigenous cultures maintain deep spiritual connections to particular species and ecosystems, with biodiversity loss representing not just environmental degradation, but cultural impoverishment. The stability and resilience of ecosystem services depend critically on maintaining the biodiversity that provides them. Simplified ecosystems may continue to provide some services in the short term, but they lack the redundancy and adaptive capacity needed to maintain service provision under changing conditions. As we explore the intricate evolutionary relationships that have shaped these service providing communities, we begin to understand how millions of years of coevolution have created the finely tuned biological partnerships that make ecosystem services possible. In the intricate ballet of life, species do not evolve in isolation. Instead, they engage in an endless dance of mutual influence, each partner's steps shaped by the movements of the other. This process, known as coevolution, has choreographed some of the most remarkable partnerships and rivalries in the natural world. Over millions of years, these evolutionary relationships have woven the complex tapestry of dependencies that characterize biodiverse ecosystems, creating intricate connections that bind species together in ways both beautiful and essential. The concept of coevolution emerged from the recognition that evolutionary change in one species can create selective pressures that drive evolutionary responses in other species, which in turn influence the evolution of the first species. This reciprocal evolutionary influence creates feedback loops that can lead to extraordinary specialization and interdependence between species. Unlike simple adaptation to physical environments, coevolution involves living organisms adapting to other living organisms that are themselves evolving in response. Perhaps nowhere is coevolution more elegantly displayed than in the relationships between flowering plants and their pollinators. The very existence of flowers represents one of evolution's most successful experiments in advertising and mutual benefit. Over 100 million years ago, as the first flowering plants began to diversify, they entered into partnerships with insects, birds, and other animals that would fundamentally transform both groups and ultimately reshape terrestrial ecosystems. Consider the relationship between yucca plants and yucca moths, one of the most specialized examples of coevolution known to science. Each species of yucca plant has its own dedicated species of moth, and neither can survive without the other. The female moth collects pollen from yucca flowers, forms it into a ball, and carries it to another flower where she deposits her eggs inside the flower's ovary. She then deliberately places the pollen ball on the flower's stigma, ensuring that the plant will be fertilized and produce seeds. The moth's larvae feed on some of these developing seeds, but enough remain to ensure the plant's reproduction. This partnership has evolved to such precision that the timing of moth emergence perfectly matches the blooming period of their specific yucca species. The moth's mouthparts have evolved specifically for collecting and manipulating yucca pollen, while the yucca flowers have evolved features that facilitate the moth's pollination behavior. Neither species could survive the loss of its partner, creating an evolutionary bond that has persisted for millions of years. Orchids and their pollinators showcase another dimension of plant-pollinator coevolution, one that involves deception as well as mutual benefit. Many orchid species have evolved elaborate mechanisms to attract specific pollinators without providing any reward. Some orchids mimic the appearance and scent of female insects so convincingly that male insects attempt to mate with the flowers, inadvertently picking up pollen in the process. Others produce chemical compounds that mimic the alarm pheromones of bees, attracting aggressive males who attempt to attack the perceived intruder. The precision required for these deceptive strategies reveals the power of coevolutionary processes. The orchids must evolve to match not just the visual appearance of their target species, but also their chemical signals and even their texture. Meanwhile, the deceived pollinators face selective pressure to avoid being fooled, leading to an ongoing evolutionary arms race between deceiver and deceived. Coevolution between predators and prey has produced some of the most dramatic examples of evolutionary escalation in Tween Question 1. As predators evolve more effective hunting strategies, their prey evolve better defenses, which in turn select for improved predation abilities, creating cycles of adaptation and counter-adaptation that can continue for millions of years. The relationship between cheetahs and their prey illustrates this dynamic clearly. Cheetahs have evolved extraordinary speed, reaching 70 miles per hour in pursuit of antelope and other fast-running prey. But their prey has not remained static. Gazelles and other antelope species have evolved not just speed, but also agility, endurance, and sophisticated predator detection systems. Young gazelles practice their escape maneuvers through play, honing the skills that may someday save their lives. This predator-prey arms race has shaped both participants in profound ways. Cheetahs have sacrificed strength and climbing ability for speed, becoming the most specialized of the large cats. Their lightweight build and non-retractable claws make them exceptional sprinters, but poor fighters, leaving them vulnerable to other predators. Meanwhile, their prey species have evolved group behaviors, vigilance patterns, and escape strategies specifically tailored to avoiding cheetah predation. Plant-herbivore coevolution has created an extraordinary diversity of chemical warfare strategies. Plants, unable to run from their predators, have evolved an arsenal of toxic compounds, digestibility reducers, and other chemical defenses. In response, herbivores have evolved detoxification systems, behavioral avoidances, and even the ability to turn plant toxins to their own advantage. The relationship between milkweeds and monarch butterflies exemplifies how herbivores can turn plant defenses into their own protection. Milkweeds produce toxic cardiac glycosides that can kill most herbivores, and milkweeds produce toxic cardiac glycosides. Monarch caterpillars, however, have evolved the ability to sequester these toxins in their tissues without being harmed. The toxins persist through metamorphosis, making adult monarchs toxic to bird predators. The monarch's bright orange coloration serves as a warning signal, advertising their toxicity to potential predators. This relationship has driven further coevolutionary responses. Some bird species have learned to eat monarchs despite their toxicity, either by avoiding the most toxic body parts or by developing tolerance to the cardiac glycosides. In response, monarchs have evolved behavioral strategies, such as overwintering in dense aggregations, where their collective warning signals are more effective. Coevolution has also shaped some of the most intimate biological relationships, including the many forms of symbiosis that characterize complex ecosystems. The relationship between flowering plants and mycorrhizal fungi represents one of the most widespread and ancient of these partnerships. Most plant species form associations with soil fungi that extend their root systems and help them access nutrients from greater volumes of soil. In return, plants provide fungi with carbohydrates produced through photosynthesis. This partnership has co-evolved over hundreds of millions of years, becoming so fundamental that most plants cannot survive without their fungal partners. The fungi have evolved specialized structures for interfacing with plant roots, while plants have evolved mechanisms for recognizing and establishing relationships with beneficial fungi. Some plant families have co-evolved such intimate relationships with specific groups of fungi that they form exclusive partnerships found nowhere else in nature. The coevolution of legume plants and nitrogen-fixing bacteria provides another example of intimate biological partnership. These bacteria, housed in specialized root nodules, convert atmospheric nitrogen into forms that plants can use, providing an essential nutrient that is often limiting in terrestrial ecosystems. In return, the plants provide the bacteria with carbohydrates and a protected environment. This relationship has shaped the evolution of both partners in remarkable ways. The bacteria have evolved complex communication systems that allow them to recognize compatible host plants and negotiate the terms of their partnership. Plants have evolved sophisticated mechanisms for controlling their bacterial partners, including the ability to reduce nutrient flow to nodules that are not performing well. This biological market economy ensures that both partners benefit from the relationship. Coevolutionary relationships often involve more than two species, creating complex networks of evolutionary interdependence. The relationship between figs and fig wasps involves not just the mutualistic wasps that pollinate the figs, but also parasitic wasps that exploit the system, predators that feed on the wasps, and other species that depend on figs for food. Each species in this network influences the evolution of the others, creating a web of coevolutionary relationships that can involve dozens of species. These multi-species coevolutionary networks help explain why biodiversity loss can have such far-reaching consequences. When one species disappears from a co-evolved network, it can disrupt the evolutionary relationships that other species depend on. The loss of a key pollinator may lead to the decline of its plant partners, which in turn affects the herbivores, predators, and decomposers that depend on those plants. Understanding coevolution reveals why biodiversity conservation requires an ecosystem-level approach. Protecting individual species is not enough if the coevolutionary relationships that sustain them are disrupted. Maintaining the complex networks of interaction that characterize biodiverse ecosystems requires preserving entire communities of co-evolved species and the habitats that support their intricate relationships. As we explore the pathways through which energy and materials flow among these co-evolved partners, we begin to see how their evolutionary relationships create the complex food webs that sustain ecosystem function and provide the foundation for all higher levels of biological organization. In the grand economy of nature, energy and nutrients flow through intricate networks that connect every living thing in webs of dependence and exchange. Unlike the linear food chains depicted in elementary textbooks, real ecosystems operate through complex food webs where energy and materials travel along multiple pathways, creating redundancies and interconnections that provide stability in an inherently uncertain world. These webs represent the circulatory systems of ecosystems, moving the essential resources that sustain life, while linking species across vast differences in size, habitat, and lifestyle. At the foundation of every food web lies the fundamental law of thermodynamics. Energy cannot be created or destroyed, only transformed from one form to another. Solar energy, captured by photosynthetic organisms, provides the currency that drives terrestrial ecosystems, while in deep ocean environments, chemical energy from hydrothermal vents or decomposing organic matter serves similar functions. This energy flows through ecosystems in predictable patterns, diminishing at each transfer, but creating the energetic foundation for all biological activity. Primary producers form the base of food webs, converting inorganic materials into the organic molecules that fuel ecosystem function. But even at this foundational level, the complexity of real food webs becomes apparent. Different producer species operate most efficiently under different conditions, creating temporal and spatial mosaics of primary production. In temperate forests, spring ephemeral wildflowers capture sunlight before tree leaves emerge, while shade tolerant understory plants photosynthesize throughout the growing season. Aquatic phytoplankton bloom at different times and in different locations based on nutrient availability and water temperature. This diversity among primary producers creates multiple entry points for energy to enter food webs, providing stability against environmental fluctuations. When drought stress reduces tree photosynthesis, grasses and other drought tolerant plants may continue producing organic matter. When changing water temperatures affect one group of phytoplankton, others adapted to different thermal conditions can maintain primary production. Primary consumers occupy the next level of food webs, but their feeding relationships reveal the first hints of the complexity that characterizes natural systems. Herbivores rarely consume plants indiscriminately. Instead, they exhibit preferences based on nutritional content, digestibility, toxicity, and accessibility. These feeding preferences create different patterns of energy flow depending on which herbivore species are present and abundant. Large grazing mammals like bison and zebras focus primarily on grasses, creating one pathway for energy flow from primary producers to higher trophic levels. Smaller mammals like rabbits and rodents may consume a broader variety of plants, including seeds, fruits, and tender shoots. Flying herbivores, such as many insects and birds, can access plant parts unavailable to ground dwelling mammals, creating additional pathways for energy transfer. The seasonal timing of herbivore feeding adds another layer of complexity to food web dynamics. Some herbivores concentrate their feeding during brief periods when their preferred foods are most abundant and nutritious. Others spread their feeding throughout the year, switching among different food sources as availability changes. These temporal patterns influence not just the herbivores themselves, but the predators that depend on them and the plants that serve as their food sources. Secondary consumers introduce the predator prey dynamics that add both stability and complexity to food web structure. But predation itself encompasses a remarkable diversity of strategies and target species. Some predators specialize in particular types of prey, becoming tightly linked to the population dynamics of their food sources. Others operate as generalists, switching among different prey species as opportunities arise. The presence of both specialist and generalist predators creates different types of stability within food webs. Specialist predators can effectively control the populations of their preferred prey, preventing any single herbivore species from overwhelming plant communities. However, specialists remain vulnerable to crashes in their prey populations. Generalist predators provide a different type of stability, serving as a buffer that can absorb fluctuations in any single prey species while maintaining predation pressure across the entire herbivore community. Intraguild predation, where predators also prey upon each other, adds another dimension to food web complexity. Large predators often consume smaller predators, creating multiple pathways for energy flow and complex indirect effects throughout the food web. When wolves kill coyotes, they not only reduce coyote populations directly, but also indirectly benefit the small mammals that coyotes would have consumed. These cascading effects can influence plant communities if the released small mammals increase their herbivory pressure. Decomposer pathways represent one of the most important yet often overlooked components of food webs. Dead organic matter from all trophic levels provides energy and nutrients for communities of bacteria, fungi, and invertebrates that break down complex molecules and return nutrients to forms that primary producers can use. This decomposer food web operates largely independently of the more visible grazing food web, yet it processes the majority of primary production in most ecosystems. The efficiency of decomposer pathways influences the entire structure of food webs. In ecosystems with rapid decomposition, nutrients cycle quickly and remain available to support continued primary production. In systems with slow decomposition, nutrients become tied up in organic matter, potentially limiting primary production and constraining the energy available to support higher trophic levels. Detritus-based food chains create additional complexity in aquatic ecosystems, where dead organic matter from terrestrial environments provides a major energy source. Leaves falling into streams, for example, support communities of shredding insects that break down the organic matter and make it available to filtering organisms downstream. This terrestrial subsidy can be so important that removing riparian forests dramatically reduces the productivity of stream ecosystems. The concept of trophic cascades reveals how changes at one level of a food web can propagate throughout the entire system. The classic example comes from marine kelp forests, where sea otters control populations of sea urchins that graze on kelp. When otter populations decline, urchin populations explode and overgraze the kelp, transforming productive kelp forests into barren landscapes. The loss of kelp affects not just the urchins and otters, but all the fish, invertebrates, and other organisms that depend on kelp forests for habitat and food. Trophic cascades can operate through multiple pathways simultaneously. In Yellowstone National Park, the reintroduction of wolves influenced elk behavior as well as elk numbers. Elk avoided areas where they felt vulnerable to wolf predation, reducing browsing pressure on willows and aspens in those locations. The recovery of these woody plants altered stream morphology, provided habitat for songbirds, and supported beaver populations that further modified aquatic habitats. The strength of trophic cascades depends on the diversity and complexity of food web connections. In simple food webs with few alternative pathways, the loss of a key predator can have dramatic effects throughout the system. In more complex webs with multiple predators and abundant alternative food sources, the effects of losing any single species may be buffered by the presence of other species that can fulfill similar roles. Omnivory, where organisms feed at multiple trophic levels, adds stability to food webs by creating additional pathways for energy flow. Bears exemplify this strategy, feeding on plant materials, insects, fish, and mammals, depending on seasonal availability. This flexible feeding behavior allows omnivores to persist through fluctuations in any single food source while maintaining their roles in food web dynamics. The spatial structure of food webs becomes important when considering how energy and nutrients move across landscapes. Many organisms move between different habitats during their life cycles, creating connections between food webs that might otherwise remain separate. Salmon connect marine and freshwater food webs by feeding in the ocean and then returning to spawn in streams, bringing marine-derived nutrients to terrestrial ecosystems. Migratory birds link food webs across continents, moving nutrients and energy between breeding and wintering grounds. Understanding food web complexity helps explain why biodiversity loss can have such far reaching effects. Each species represents a node in the food web connected to multiple other species through feeding relationships. The loss of a single species can disrupt multiple pathways for energy and nutrient flow, potentially destabilizing the entire web. However, the presence of alternative pathways in diverse food webs provides resilience against such disruptions. As we examine the mechanisms that allow biodiverse ecosystems to maintain stability despite constant environmental fluctuations, we begin to see how the complexity of food webs contributes to the remarkable resilience that characterizes healthy natural systems. In the quiet moments before dawn, as the world balances between night and day, ecosystems everywhere demonstrate one of nature's most remarkable achievements: the ability to maintain stability in the face of constant change. Storm systems sweep across landscapes, seasons cycle through their ancient rhythms, and countless organisms live out their individual dramas of birth, growth, and death. Yet, through all this flux, healthy ecosystems persist, maintaining their essential character and function despite the chaos that surrounds and permeates them. This stability emerges not from rigidity, but from flexibility. Like a reed that bends in the wind rather than breaking, biodiverse ecosystems possess an inherent resilience that allows them to absorb disturbances, adapt to changing conditions, and recover from setbacks. Understanding this resilience reveals why diversity matters so profoundly for the long term health of our planet's life support systems. Ecological stability encompasses several related but distinct concepts that help us understand how ecosystems maintain their integrity over time. Resistance refers to an ecosystem's ability to remain unchanged in the face of disturbance. Some systems, particularly those adapted to harsh or highly variable environments, have evolved remarkable resistance to specific types of disturbance. Desert ecosystems, for example, show little change during drought periods that would devastate temperate forests. Resilience, in contrast, measures how quickly an ecosystem returns to its original state after being disturbed. A prairie fire may temporarily eliminate above ground vegetation, but the extensive root systems of grassland plants allow rapid regrowth once conditions become favorable. The ecosystem may look devastated immediately after the fire, but its resilience ensures recovery within a single growing season. Stability encompasses both resistance and resilience, describing an ecosystem's overall ability to maintain its essential characteristics through time. Stable ecosystems may fluctuate within normal bounds, but they retain their fundamental structure and function. A forest may vary in the exact composition of tree species from year to year, but it remains recognizably a forest rather than transforming into a grassland or desert. Biodiversity contributes to stability through several mechanisms that operate at different scales and time frames. Functional redundancy provides one of the most important forms of insurance against environmental uncertainty. When multiple species perform similar ecological roles, the loss of any single species is less likely to disrupt ecosystem function. If one pollinator species declines, others may be able to maintain pollination services. If a particular predator disappears, other predators may increase their consumption of the same prey species. The insurance value of functional redundancy becomes apparent during environmental stress. Different species within the same functional group often respond differently to environmental challenges. Some may be more tolerant of drought, others more resistant to disease, still others better adapted to temperature fluctuations. This response diversity ensures that some members of each functional group can maintain their ecological roles even when conditions become unfavorable for others. In grassland ecosystems, the presence of multiple grass species with different rooting depths, growing seasons, and drought tolerances provides stability against climatic variation. During wet years, shallow-rooted species may dominate, taking advantage of abundant surface moisture. During dry years, deep-rooted species access groundwater and maintain primary production when shallow-rooted species become dormant. The overall productivity of the grassland remains more stable than would be possible with any single species alone. The portfolio effect operates similarly to financial diversification, spreading ecological risk across multiple species and functional groups. Just as a diverse investment portfolio reduces financial risk by not depending entirely on any single stock or sector, biodiverse ecosystems reduce ecological risk by distributing essential functions across multiple species. When environmental conditions favor some species while challenging others, the overall ecosystem function remains more stable than would be possible in a simplified system. Temporal complementarity adds another dimension to ecosystem stability by ensuring that essential functions are maintained throughout different seasons and environmental conditions. In temperate forests, different tree species leaf out at different times in spring, extending the overall period of canopy photosynthesis. Some understory plants complete their life cycles in early spring before tree leaves block sunlight, while others photosynthesize throughout the summer using the reduced light that penetrates the canopy. This temporal partitioning of resources and functions means that ecosystem processes continue throughout changing conditions. As some species become less active during unfavorable periods, others increase their activity, maintaining the overall flow of energy and nutrients through the system. The result is greater stability in ecosystem function than would be achieved if all species operated on the same seasonal schedule. Spatial heterogeneity within biodiverse ecosystems creates additional sources of stability. Different microhabitats support different assemblages of species, creating a mosaic of conditions across the landscape. When disturbance affects part of this mosaic, unaffected areas serve as sources of organisms to recolonize damaged sites. The greater the diversity of microhabitats and the species they support, the more likely it becomes that some portions of the ecosystem will survive any particular disturbance. Forest ecosystems demonstrate this principle clearly. A diverse forest contains trees of different ages, sizes, and species distributed across various topographic positions and soil types. When a windstorm creates gaps in the canopy, different species respond in different ways. Some existing understory plants rapidly expand into the newly available light. Seeds of pioneer species, perhaps dormant in the soil for years, germinate and establish in the disturbed areas. Surviving mature trees around gap edges provide seed sources for forest regeneration. The complexity of species interactions in biodiverse systems creates additional sources of stability through negative feedback loops that prevent any single species from dominating. As the population of one species increases, its predators, parasites, and competitors typically also increase, creating pressures that limit further population growth. These density-dependent processes help maintain the relative abundance of different species within bounds that preserve ecosystem function. Keystone species play particularly important roles in maintaining ecosystem stability, often through their effects on community structure and species interactions. Sea otters in kelp forest ecosystems control sea urchin populations that would otherwise overgraze the kelp. The presence of otters maintains the complex three-dimensional habitat that supports numerous other species. Without this keystone species, the entire ecosystem can shift to an alternative stable state, dominated by urchins and characterized by much lower diversity and productivity. The loss of keystone species can trigger trophic cascades that fundamentally alter ecosystem structure and function. However, in highly diverse systems, the effects of losing any single species may be buffered by the presence of other species that can partially compensate for the lost functions. The more diverse the system, the more likely it becomes that alternative species can maintain essential ecosystem processes. Habitat connectivity enhances stability by facilitating the movement of organisms among different areas within a landscape. When local populations are stressed or eliminated by disturbance, immigration from nearby populations can speed recovery. Genetic exchange among populations maintains the genetic diversity that provides raw material for adaptation to changing conditions. Mobile species can track changing environmental conditions by moving among different habitats, maintaining their populations while contributing to ecosystem function across the landscape. Climate change provides a particularly challenging test of ecosystem stability as it represents a directional change rather than the fluctuating conditions to which ecosystems have adapted. However, biodiverse systems appear better able to cope with climate change than simplified systems. The presence of multiple species with different thermal tolerances and climatic requirements increases the likelihood that some species will be able to persist under new conditions. Genetic diversity within species provides the variation necessary for evolutionary adaptation to changing climate. The stability provided by biodiversity becomes especially apparent when systems lose their diversity. Agricultural monocultures, for example, lack the functional redundancy and response diversity of natural ecosystems. They remain highly productive under optimal conditions, but become extremely vulnerable to pests, diseases, and environmental stress. The Irish potato famine of the 1840s dramatically illustrated this vulnerability, as the genetic uniformity of potato crops made them susceptible to a single pathogen that destroyed much of the harvest for several consecutive years. Understanding the relationship between diversity and stability has important implications for ecosystem management and conservation. Maintaining the full complement of species and functional groups in an ecosystem provides insurance against future environmental challenges, many of which remain unpredictable. The investment in preserving biodiversity pays dividends in the form of more stable and predictable ecosystem services. As we explore specific examples of how biodiversity functions in different types of ecosystems, we begin to see these principles of stability and resilience operating in the complex forest communities that harbor much of the world's terrestrial diversity. Step into an old-growth forest, and you enter a living cathedral where biodiversity has composed one of nature's most complex symphonies. Above, the canopy forms a vaulted ceiling of interwoven branches, while beneath your feet, countless organisms work in the shadows to maintain the intricate processes that sustain this towering community. Every surface, from bark to leaf to fallen log, hosts its own assemblage of specialists. The air itself carries spores, pollen, and chemical messages that coordinate the activities of organisms separated by hundreds of feet and vastly different ways of life. Forests represent perhaps the most structurally complex terrestrial ecosystems, their vertical architecture creating multiple worlds stacked one upon another. This three-dimensional complexity provides the physical foundation for extraordinary biodiversity, while the web of interactions among forest species creates emergent properties that transform collections of individual trees into integrated ecosystems capable of modifying regional climate, cycling vast quantities of nutrients, and supporting communities of staggering richness. The forest canopy, that aerial realm of sunlight and wind, harbors communities so distinct from those on the forest floor that early botanists considered them different ecosystems entirely. Here, epiphytes, plants that grow on other plants without parasitizing them, create gardens in the sky. In tropical rainforests, a single large tree may support hundreds of epiphyte species, including orchids, bromeliads, ferns, and mosses that have never touched the ground. These aerial gardens capture nutrients from rainfall, dust, and decomposing organic matter, creating soil-filled microecosystems suspended high above the earth. The canopy's three-dimensional complexity provides countless microhabitats with different exposures to light, wind, and moisture. The outer canopy experiences full sunlight and dramatic temperature fluctuations, while inner branches remain buffered in more stable conditions. This environmental heterogeneity allows species with different tolerances and requirements to coexist within the same tree, multiplying the number of ecological niches available within the forest. Canopy animals have evolved remarkable adaptations for life in this aerial world. Many have developed enhanced climbing abilities, prehensile tails, or gliding membranes that allow them to navigate through the three-dimensional maze of branches. Some primates spend their entire lives in the canopy, developing specialized locomotion techniques and social behaviors adapted to their arboreal lifestyle. Birds exploit different portions of the canopy architecture, with some species feeding in the outer sun-dappled foliage, while others hunt in the shadows of interior branches. The understory creates yet another distinct world beneath the main canopy, characterized by filtered light and protection from wind and temperature extremes. Plants in this layer have evolved broad, thin leaves to capture the limited light that penetrates the canopy, often developing distinctive, deep green coloration that maximizes their light harvesting efficiency. Many understory plants exhibit remarkable phenotypic plasticity, adjusting their leaf size, shape, and orientation in response to changing light conditions as gaps open and close in the canopy above. This filtered light environment supports communities of animals adapted to low light conditions and limited flying space. Many understory birds have evolved enhanced hearing and vocalizations for communicating through dense vegetation. Forest mammals often develop keen senses of smell and touch to navigate and find food in dim conditions. Insects in the understory frequently display cryptic coloration that makes them nearly invisible against bark and leaves. The forest floor represents the foundation layer where decomposition processes drive nutrient cycling throughout the entire ecosystem. Here, fallen leaves, branches, and other organic matter create a constantly changing mosaic of microhabitats. Recent leaf fall provides fresh organic matter that supports different decomposer communities than older, more decomposed material. Fallen logs in various stages of decay create distinct microenvironments with different moisture levels, nutrient concentrations, and microbial communities. This detrital layer hosts an extraordinary diversity of decomposer organisms, from bacteria and fungi to countless invertebrates that specialize in breaking down different types of organic matter. Some species focus on fresh leaf litter, others on partially decomposed material, and still others on the highly decomposed humus that forms the soil organic layer. The succession of species involved in decomposition ensures that complex organic molecules are efficiently broken down and their nutrients return to forms that plants can absorb and use. Mycorrhizal fungi create one of the forest's most important but largely invisible networks, connecting tree roots across vast areas through underground fungal threads called hyphae. These fungi extend the effective reach of tree root systems, helping them access nutrients and water from greater volumes of soil. In return, trees provide the fungi with carbohydrates produced through photosynthesis. Some mycorrhizal networks connect multiple tree species, creating underground communication networks that can span entire forest stands. Research has revealed that these fungal networks facilitate not just nutrient exchange, but also information transfer between trees. When attacked by insects, some trees can send chemical signals through mycorrhizal networks to warn neighboring trees, which may then begin producing defensive compounds before the herbivores arrive. Mother trees have been observed transferring resources to their offspring through these networks, supporting young trees growing in their shade until they can establish their own photosynthetic capacity. The seasonal dynamics of temperate forests reveal how biodiversity creates temporal complementarity that maximizes resource use throughout the year. Spring ephemeral wildflowers like bloodroot and trout lily complete their entire above-ground life cycle in the brief window between snowmelt and canopy closure, taking advantage of the high light levels reaching the forest floor before trees leaf out. Summer understory plants have evolved shade tolerance that allows them to photosynthesize throughout the growing season using the reduced light that penetrates the full canopy. Fall brings its own suite of biological activities as trees withdraw nutrients from their leaves before dropping them, while animals prepare for winter through migration, hibernation, or food caching behaviors. Winter may seem like a time of dormancy, but many forest processes continue beneath the snow. Decomposition slows but doesn't stop, and some animals remain active, creating a network of tunnels and tracks that connects winter-active organisms across the forest floor. Forest disturbance regimes create additional dimensions of diversity through the complex mosaic of different aged forest patches across the landscape. Natural disturbances like windstorms, fires, and insect outbreaks create gaps in the forest canopy that initiate succession processes. These gaps support early successional species that require high light levels and may be rare in the mature forest. As gaps close and succession proceeds, they support different assemblages of species adapted to various stages of forest development. The gap phase regeneration cycle ensures that forests maintain species adapted to different successional stages within the same landscape. Pioneer species like aspen and birch colonize large disturbances, eventually creating conditions suitable for shade-tolerant species that characterize mature forests. This successional diversity provides habitat for animals with different requirements and maintains genetic diversity within tree populations through periods of expansion and contraction. Old-growth forests represent the culmination of forest succession, supporting biodiversity through structural features that develop only over long time periods. Large, old trees create unique habitat through their massive size, thick bark, and complex branching patterns. Dead standing trees, or snags, provide nesting sites for woodpeckers and other cavity nesting species. Large fallen logs create nurse logs that support tree regeneration while providing habitat for countless invertebrates and small vertebrates. The structural complexity of old growth forests supports species that cannot survive in younger forests. Many lichen species require the stable bark chemistry and microclimate of old trees. Certain bird species depend on the large tree cavities that develop only in ancient forests. The complex vertical structure provides numerous ecological niches unavailable in simpler forest stands. Human activities have dramatically simplified forest ecosystems in many regions, reducing the diversity of tree species, eliminating old growth structural features, and fragmenting forest landscapes. Even aged forest management creates structurally simple stands that support fewer species than natural forests, with their complex age structure and species composition. The loss of old growth forests has eliminated habitat for specialized species while reducing the ecosystem services that complex forests provide. Understanding forest biodiversity reveals the importance of maintaining structural and compositional complexity for ecosystem health and resilience. Diverse forests prove more resistant to pest outbreaks, more resilient to environmental stress, and more capable of adapting to changing conditions than simplified forest monocultures. As we turn our attention to aquatic systems, we'll discover that similar principles of structural complexity and functional diversity create the foundation for biodiversity in the watery realms that cover most of our planet's surface. Beneath the surface of the world's waters lies a realm where biodiversity takes forms unimaginable on land. Here, life has evolved solutions to challenges unique to the aquatic environment, creating communities that span from microscopic plankton drifting in ocean currents to massive whales whose migrations connect ecosystems across entire ocean basins. Water itself becomes both medium and habitat, its physical properties shaping the evolution of organisms and the structure of communities in ways that create some of Earth's most diverse and productive ecosystems. The fundamental difference between terrestrial and aquatic life begins with the medium itself. Water is nearly 1,000 times denser than air, requiring different strategies for movement, support, and reproduction. Yet this density also provides advantages, offering buoyancy that allows organisms to achieve sizes impossible on land and creating three-dimensional habitats where life can exist at every level from surface to bottom. The flow of water through aquatic systems creates opportunities for filter feeding and suspension feeding that have no terrestrial equivalent, while the solvent properties of water facilitate chemical communication across vast distances. Marine ecosystems cover more than 70% of Earth's surface and contain habitats ranging from sun-drenched coral reefs to pitch-black abyssal plains, thousands of meters beneath the surface. This vast three-dimensional space encompasses the planet's largest continuous habitat, yet it remains largely unexplored, with new species discovered regularly, even in well-studied regions. The ocean's biodiversity patterns reflect the complex interplay of light, temperature, nutrients, and currents that create distinct zones and regions, each supporting characteristic assemblages of organisms. Near the ocean's surface, the photic zone buzzes with microscopic life that forms the foundation of marine food webs. Phytoplankton, including both single-celled algae and photosynthetic bacteria, convert sunlight and dissolve nutrients into organic matter that supports virtually all marine life. These microscopic producers exhibit extraordinary diversity, with different species adapted to varying light levels, nutrient concentrations, and water temperatures. Some phytoplankton form elaborate glass houses called frustules, while others develop intricate calcium carbonate plates or complex organic cell walls. The seasonal and spatial patterns of phytoplankton blooms create pulses of productivity that ripple through entire marine food webs. In polar regions, the brief summer period of continuous daylight triggers massive algal blooms that support enormous populations of krill and other zooplankton. These blooms, in turn, attract baleen whales that undertake epic migrations to feast on the concentrated food sources. The timing of these blooms has evolved to match the life cycles of the animals that depend on them, creating intricate temporal synchronizations that connect organisms across multiple trophic levels. Zooplankton communities add another layer of complexity to pelagic ecosystems, encompassing organisms from microscopic protozoans to jellyfish several meters across. Many zooplankton undertake daily vertical migrations, rising toward the surface to feed at night and descending to deeper waters during the day. This behavior, repeated by millions of organisms, creates one of the largest daily movements of biomass on the planet and facilitates the transport of nutrients between different ocean layers. The diversity of zooplankton feeding strategies reflects the variety of food sources available in the open ocean. Some species graze on phytoplankton like underwater cows, while others hunt other zooplankton with sophisticated capture mechanisms. Copepods use feathery appendages to create feeding currents that draw small particles toward their mouths. Jellyfish deploy stinging tentacles to capture prey, while comb jellies use sticky cells to ensnare victims. This diversity of feeding mechanisms ensures that different size classes and types of food are efficiently harvested from the water column. Coral reef ecosystems represent marine biodiversity hotspots where exceptional productivity and structural complexity support communities rivaling tropical rainforests in their species richness. The three-dimensional calcium carbonate framework created by coral colonies provides countless microhabitats with different exposures to light, current, and predation pressure. Different coral species dominate at different depths and exposures, creating a complex mosaic of habitat types within individual reef systems. The partnership between corals and their symbiotic algae, zooxanthellae, demonstrates one of the most important mutualistic relationships in marine ecosystems. These microscopic algae live within coral tissues, using coral waste products and sunlight to produce organic compounds that they share with their hosts. This relationship allows corals to thrive in nutrient-poor tropical waters by essentially cultivating their own food source. The diversity of zooxanthellae species and their different environmental tolerances contributes to the ability of coral reefs to persist across varying conditions. Fish communities on coral reefs exhibit extraordinary diversity in both species composition and ecological roles. Cleaner fish establish cleaning stations where larger fish come to have parasites removed, creating mutualistic networks that help maintain fish health across the reef community. Herbivorous fish control algal growth that might otherwise overgrow corals, while predatory fish regulate the populations of herbivores and invertebrates. The complex territorial behaviors and feeding strategies of reef fish create intricate spatial patterns of resource use that allow dozens of species to coexist within small areas. Deep sea environments, once thought to be biological deserts, harbor communities of surprising diversity adapted to extreme conditions of darkness, cold, and pressure. The absence of photosynthesis in these aphotic zones means that deep sea communities depend on the rain of organic matter from surface waters or on chemosynthetic bacteria that derive energy from chemical reactions. Hydrothermal vents create oases of productivity in the deep ocean, supporting communities based entirely on chemosynthesis and including many species found nowhere else on Earth. The adaptations of deep sea organisms reveal the creative solutions that evolution finds for extreme environments. Many deep sea fish have evolved bioluminescence, creating their own light through chemical reactions. Some species use light as a lure to attract prey, while others employ it for communication or predator confusion. The absence of light has led to the evolution of enhanced sensory systems, with many deep sea organisms developing extraordinary sensitivity to chemical cues, pressure changes, and water movements. Freshwater ecosystems, though covering less than 1% of Earth's surface, support disproportionate numbers of species relative to their area. Rivers, lakes, and wetlands serve as biodiversity hotspots for many groups of organisms, particularly fish, mollusks, and aquatic insects. The isolation of freshwater systems creates opportunities for endemic species to evolve, leading to high levels of localized diversity that contribute significantly to global biodiversity. Rivers and streams create linear habitats that connect terrestrial and aquatic ecosystems while supporting specialized communities adapted to flowing water. The continuum from headwater streams to large rivers creates a gradient of habitat conditions that supports different assemblages of organisms. Cold, fast-flowing headwater streams support communities adapted to high oxygen levels and rocky substrates, while warm, slow-flowing lowland rivers support different communities adapted to fine sediments and lower oxygen levels. The longitudinal connectivity of river systems allows some species to complete complex life cycles that span multiple habitat types. Salmon undertake epic migrations between marine and freshwater environments, spending their adult lives feeding in the ocean before returning to natal streams to reproduce. These migrations transport marine-derived nutrients into terrestrial ecosystems, with spawning salmon providing food for bears, eagles, and other terrestrial predators, while their decomposing carcasses fertilize riparian forests. Lake ecosystems support biodiversity through their complex three-dimensional structure and seasonal cycles. Thermal stratification in deep lakes creates distinct water layers with different temperatures, oxygen levels, and nutrient concentrations. Different species occupy these layers, creating vertical zonation patterns similar to those seen in forests. The seasonal mixing of lake waters redistributes nutrients and oxygen, triggering biological responses that connect different components of lake food webs. Ancient lakes, like Lake Baikal and the African Rift Valley lakes, have served as evolutionary laboratories where endemic species have proliferated over millions of years. Lake Baikal alone contains over 1,700 endemic species, including the world's only freshwater seal and numerous endemic fish, invertebrates, and microorganisms. These ancient systems demonstrate how geographic isolation and stable environmental conditions can lead to extraordinary levels of endemism and evolutionary innovation. Wetland ecosystems create transitional zones between terrestrial and aquatic environments that support specialized communities adapted to fluctuating water levels. The periodic drying and flooding of wetlands creates complex habitat mosaics that support different species during different phases of the hydrological cycle. Many wetland species have evolved strategies for surviving both aquatic and terrestrial conditions, while others time their life cycles to take advantage of specific water level conditions. The loss and degradation of aquatic ecosystems pose particular challenges for biodiversity conservation because freshwater and marine systems often serve as repositories for pollutants and nutrients from surrounding landscapes. Aquatic organisms frequently face multiple stressors, including habitat loss, pollution, overharvesting, and climate change. The high connectivity of aquatic systems means that impacts in one area can affect organisms throughout entire watersheds or ocean basins. Understanding aquatic biodiversity reveals the critical importance of maintaining water quality and habitat connectivity for preserving these remarkable communities. As we explore the often overlooked world of microbial diversity, we begin to appreciate how these tiny organisms form the foundation that supports all the larger forms of life we see in both aquatic and terrestrial ecosystems. In every teaspoon of soil beneath your feet live more microorganisms than there are people on Earth. In the depths of the ocean, in the ice of polar glaciers, in the scalding waters of hot springs, and even within your own body, microscopic life forms carry out the fundamental processes that make all other life possible. These invisible communities represent the vast majority of life's diversity, encompassing billions of species that remain largely unknown to science, yet whose collective activities regulate the chemistry of our atmosphere, cycle essential nutrients through ecosystems, and maintain the conditions necessary for the existence of all visible life. Microbial diversity encompasses three entire domains of life: bacteria, archaea, and the microscopic members of the eukaryotic domain that includes protists and single-celled fungi. This represents the deepest branches of life's evolutionary tree, spanning nearly 4 billion years of biological innovation. While plants and animals represent recent additions to life's story, microorganisms have been experimenting with different ways of living for most of Earth's history. Evolving biochemical pathways and ecological strategies that form the foundation of all modern ecosystems. The metabolic diversity of microorganisms dwarfs that of all other life forms combined. While plants and animals rely on a relatively small number of biochemical pathways to generate energy and build cellular components, microorganisms have evolved countless alternatives that allow them to thrive in environments where no other life can survive. Some bacteria derive energy from hydrogen sulfide, others from methane, iron, or even radioactive decay. These diverse metabolic strategies have made microorganisms the ultimate pioneers, colonizing every conceivable habitat on Earth and establishing the biogeochemical cycles that sustain larger forms of life. Photosynthetic bacteria were Earth's first solar energy converters, releasing oxygen as a waste product billions of years before the first plants evolved. These ancient cyanobacteria fundamentally transformed Earth's atmosphere, creating the oxygen-rich conditions that eventually allowed complex multicellular life to evolve. Today, marine cyanobacteria and other photosynthetic microorganisms continue to produce much of the oxygen we breathe and serve as the foundation of oceanic food webs. The nitrogen cycle, essential for all life because nitrogen is a critical component of proteins and nucleic acids, depends almost entirely on microbial processes. Nitrogen fixing bacteria convert atmospheric nitrogen gas into ammonia that plants can absorb and use. Without these microorganisms, the vast reservoirs of nitrogen gas in the atmosphere would remain inaccessible to most life forms. Other specialized bacteria convert organic nitrogen compounds back to nitrogen gas, completing the cycle and preventing the accumulation of nitrogen compounds that could become toxic to many organisms. In terrestrial ecosystems, soil microorganisms create the foundation for plant life through their role in nutrient cycling and soil formation. Bacteria and fungi decompose organic matter, breaking down complex molecules and releasing nutrients in forms that plants can absorb. The diversity of decomposer microorganisms ensures that different types of organic matter are efficiently processed, from simple sugars to complex lignins and celluloses that make up woody plant tissues. Mycorrhizal fungi form one of the most widespread and important symbiotic relationships on Earth, partnering with the roots of most plant species. These fungi extend the effective reach of plant root systems, helping plants access nutrients and water from greater volumes of soil. In return, plants provide fungi with carbohydrates produced through photosynthesis. This ancient partnership, which evolved over 400 million years ago, has been crucial for the colonization of terrestrial environments by plants and continues to support forest and grassland ecosystems worldwide. The networks formed by mycorrhizal fungi create underground communication systems that connect individual plants across landscapes. Research has revealed that these fungal networks can transport not just nutrients, but also chemical signals that warn of insect attacks or other environmental stresses. Some networks span entire forest stands, connecting trees of different species and ages in vast underground webs that help coordinate ecosystem responses to environmental change. Aquatic ecosystems depend equally heavily on microbial diversity, though the organisms and processes involved often differ from their terrestrial counterparts. Marine bacteria play crucial roles in processing the vast amounts of organic matter produced by phytoplankton, breaking down complex molecules and recycling nutrients that support continued primary production. Some marine bacteria have evolved the ability to break down pollutants, helping to clean up oil spills and other contamination. In the deep ocean, chemosynthetic bacteria create oases of life around hydrothermal vents and cold seeps where sunlight cannot penetrate. These bacteria derive energy from chemical reactions involving hydrogen sulfide, methane, or other compounds, supporting entire ecosystems independent of photosynthesis. The discovery of these chemosynthetic ecosystems revolutionized our understanding of life's possibilities and expanded the range of environments where life might exist on other planets. Microbial communities in extreme environments have revealed the remarkable limits of life on Earth. Thermophilic bacteria thrive in hot springs and hydrothermal vents at temperatures that would cook most organisms. Psychrophilic bacteria remain active in polar ice and deep ocean waters near freezing. Halophilic archaea survive in salt concentrations that would quickly kill other organisms, while acidophilic bacteria flourish in acidic mine drainage with pH levels comparable to battery acid. These extremophiles not only expand our understanding of life's limits, but also provide insights into the conditions under which life first evolved on Earth. Many extreme environments resemble the harsh conditions that existed on early Earth, suggesting that life may have originated in what we now consider extreme environments. The biochemical innovations of extremophiles also provide inspiration for biotechnology applications, from enzymes that function at high temperatures to organisms that can remediate environmental contamination. The human microbiome illustrates how microbial diversity directly impacts our daily lives and health. Each person hosts trillions of microorganisms, representing thousands of species that live on our skin, in our mouths, and throughout our digestive systems. These microbial communities help digest food, produce vitamins, protect against pathogens, and regulate immune system function. The composition of our microbiome influences everything from our susceptibility to infections to our mental health and metabolism. Research into the human microbiome has revealed that maintaining microbial diversity within our bodies is crucial for health. People with more diverse gut microbiomes generally exhibit better health outcomes and greater resistance to disease. Disruptions to microbial diversity, often caused by antibiotic use or poor diet, can lead to increased susceptibility to infections, autoimmune disorders, and metabolic diseases. This understanding has led to growing interest in probiotics, fecal microbiome transplants, and other therapies designed to restore healthy microbial communities. Agricultural ecosystems depend heavily on soil microbial diversity for productivity and sustainability. Healthy agricultural soils contain vast communities of bacteria, fungi, protozoa, and other microorganisms that cycle nutrients, suppress plant diseases, and maintain soil structure. Industrial agriculture practices that rely heavily on synthetic fertilizers and pesticides often reduce soil microbial diversity, leading to decreased soil health and increased dependence on external inputs. Sustainable agricultural practices that maintain soil microbial diversity can improve crop yields while reducing environmental impacts. Cover cropping, reduced tillage, and organic matter additions support diverse microbial communities that provide natural fertility and pest control services. Some farmers are now inoculating crops with beneficial microorganisms to improve plant health and reduce the need for chemical inputs. Climate change has significant implications for microbial diversity and the global processes that microorganisms control. Rising temperatures may accelerate microbial decomposition of organic matter in soils, potentially releasing stored carbon and accelerating climate change. Changes in precipitation patterns could alter the composition of microbial communities, affecting nutrient cycling and ecosystem productivity. Ocean acidification, caused by increasing atmospheric carbon dioxide, may impact marine microbial communities that play crucial roles in oceanic carbon cycling. Understanding microbial responses to climate change is crucial for predicting how ecosystems will function under future conditions. Some microorganisms may adapt quickly to changing conditions due to their short generation times and large population sizes. Others may be more sensitive to environmental change, potentially disrupting the biogeochemical cycles they control. The loss of microbial diversity in any ecosystem could have cascading effects on larger organisms that depend on microbial services. Conservation efforts increasingly recognize the importance of protecting microbial diversity alongside the more visible components of biodiversity. Soil conservation practices that maintain microbial communities benefit entire ecosystems. Protecting pristine environments preserves microbial communities that may contain unknown species with potential applications for medicine, biotechnology, or environmental remediation. As we consider the unique evolutionary experiments occurring in isolated environments around the world, we begin to appreciate how geographic separation creates opportunities for both microbial and larger organisms to evolve in remarkable new directions. Imagine a seed carried by ocean winds, traveling thousands of miles before finally landing on a remote volcanic island where no plant of its kind has ever grown. Or picture a flock of birds blown off course by a powerful storm, finding themselves on an isolated mountaintop surrounded by a sea of inhospitable desert. These moments of accidental colonization have occurred countless times throughout Earth's history, creating natural laboratories where evolution experiments with new forms of life in isolation from the competitive pressures and constraints of mainland ecosystems. Geographic isolation acts as one of evolution's most powerful creative forces, providing opportunities for populations to diverge from their ancestral forms and develop characteristics found nowhere else on Earth. When populations become separated by barriers they cannot cross, whether oceans, mountain ranges, deserts, or other inhospitable terrain, they begin independent evolutionary journeys that can lead to remarkable innovations and specializations. Over time, these isolated populations may become so different from their ancestors that they represent entirely new species, genera, or even families of organisms. True oceanic islands, formed by volcanic activity far from any continental landmass, provide the clearest examples of evolution in isolation. When these islands first emerge from the ocean, they are completely sterile, lacking any form of life. Every organism that eventually colonizes them must arrive from somewhere else, crossing vast expanses of ocean through their own mobility or by chance dispersal. This filters the colonizing species to those capable of long distance travel or with propagules that can survive extended journeys through inhospitable environments. The Hawaiian Islands illustrate this process beautifully. Located over 2,000 miles from the nearest continental landmass, these volcanic islands have been colonized by organisms that arrived via three main pathways: flying under their own power, floating across the ocean, or rafting on debris. Birds, bats, and insects could fly to the islands, though the journey remained challenging enough that relatively few species made the crossing. Plant seeds and spores arrived carried by wind, ocean currents, or in the digestive systems of migrating birds. Some organisms may have rafted across on natural debris, though this pathway likely contributed few successful colonists. Once established on isolated islands, colonizing species often encounter ecological opportunities unavailable in their ancestral environments. Islands typically have fewer species than comparable mainland areas, meaning less competition for resources and unoccupied ecological niches that immigrants can exploit. Without the predators, parasites, and competitors that shape their evolution on the mainland, island colonists may be freed from selective pressures that previously constrained their evolution. The Hawaiian honeycreepers provide a classic example of adaptive radiation, the evolutionary process by which a single ancestral species gives rise to multiple descendant species that exploit different ecological niches. From a single finch-like ancestor that arrived on the Hawaiian Islands several million years ago, at least 50 species of honeycreepers evolved, each with specialized feeding behaviors and corresponding beak shapes. Some developed curved beaks for extracting nectar from flowers, others evolved robust beaks for cracking seeds, and still others developed pointed beaks for probing bark crevices for insects. This dramatic diversification occurred because the ancestral honey creepers encountered a variety of unexploited food sources on the islands. With no other species filling these ecological roles, natural selection favored individuals that could efficiently exploit different resources. Over thousands of generations, populations specializing in different foods became reproductively isolated and eventually evolved into distinct species. Madagascar represents perhaps the most spectacular example of evolution in isolation on a continental scale. This island continent separated from Africa over 100 million years ago, providing an enormous span of time for evolutionary divergence. Today, Madagascar hosts extraordinary levels of endemism, with roughly 90% of its species found nowhere else on Earth. Lemurs, which arrived on Madagascar from Africa and subsequently diversified into over 100 species, represent just one example of the island's remarkable endemic radiations. The isolation of Madagascar has allowed the evolution of ecological communities with no mainland equivalent. The island lacks many mammalian families found in Africa, such as large cats, elephants, and antelopes. In their absence, other groups have evolved to fill similar ecological roles. Some lemurs became large-bodied herbivores, while fossils reveal that Madagascar once hosted enormous flightless birds that occupied ecological niches filled elsewhere by large mammals. Island biogeography theory helps explain the patterns of diversity found on islands of different sizes and degrees of isolation. Larger islands can support more species because they contain more habitats and can sustain larger populations that are less prone to extinction. The distance from mainland source populations also influences diversity, with more isolated islands receiving fewer colonists and supporting fewer species overall. However, more isolated islands often have higher proportions of endemic species because their colonists have had more time to evolve in isolation. The concept of island biogeography extends beyond oceanic islands to any isolated habitat surrounded by inhospitable terrain. Mountain peaks separated by lowland valleys function as sky islands where alpine species become isolated and may evolve into distinct forms. Forest fragments in agricultural landscapes act as habitat islands where woodland species become separated from their relatives in other forest patches. Even lakes can serve as aquatic islands where fish and other organisms evolve in isolation from populations in other water bodies. Lake Victoria, in Africa, demonstrates how even relatively recent isolation can drive rapid evolutionary change. This massive lake, which dried up completely about 15,000 years ago, was recolonized by a small number of fish species when it refilled. One group of ancestral cichlid fish subsequently diversified into over 500 species, each adapted to different feeding strategies, water depths, and habitat types within the lake. This explosive radiation occurred in less than 15,000 years, demonstrating that evolution can operate remarkably quickly under the right conditions. The cichlids of Lake Victoria evolved diverse feeding specializations that allowed them to partition the lake's resources. Some species became algae scrapers, others evolved to eat insects, and still others became mollusk crushers or fish predators. Corresponding changes in body shape, mouth structure, and behavior accompanied these feeding specializations. The result was an intricate community of closely related species that together exploited virtually every available food source in the lake. Cave systems provide another type of isolated environment where evolution produces remarkable specializations. Many cave-dwelling organisms have evolved reduced or absent eyes, enhanced sensory systems for navigating in complete darkness, and pale coloration due to the absence of selection for pigmentation. These adaptations evolve repeatedly in different cave systems, demonstrating how similar environmental pressures produce similar evolutionary responses. The unique environmental conditions in caves select for traits rarely seen in surface environments. Without light for photosynthesis, cave ecosystems depend entirely on organic matter that enters from the outside world. This energy limitation favors organisms with extremely efficient metabolisms and behaviors that minimize energy expenditure. Some cavefish have evolved reduced metabolic rates and can survive for months without food, adaptations that would be disadvantageous in more productive surface environments. Island communities often display unusual ecological characteristics compared to their mainland counterparts. Many oceanic islands lack native terrestrial mammals, creating opportunities for other groups to evolve into ecological roles typically filled by mammals elsewhere. On some islands, birds have evolved flightlessness and increased body size, exploiting terrestrial niches normally occupied by mammals. The extinct moa of New Zealand and elephant birds of Madagascar represent extreme examples of this evolutionary trend. The absence of natural predators on many islands has led to the evolution of organisms with reduced anti-predator defenses. Island plants often lack the thorns, toxins, or other defenses common in their mainland relatives. Island animals may lose their ability to fly or develop behaviors that would make them extremely vulnerable to predators in other environments. These evolutionary changes represent adaptations to island conditions but create vulnerabilities when new species are introduced. Human activities have dramatically impacted island biodiversity, often with devastating consequences for endemic species. Islands have experienced disproportionately high rates of extinction, particularly following human colonization. The introduction of non-native predators, competitors, and diseases has eliminated many island species that evolved without defenses against these new threats. The flightless birds of New Zealand, for example, proved extremely vulnerable to introduced mammals and have suffered numerous extinctions. Climate change poses additional challenges for island biodiversity. Rising sea levels threaten low-lying islands and coastal habitats, while changing temperature and precipitation patterns may disrupt the environmental conditions to which island species have adapted. Species on isolated mountaintops face particular risks, as they may have nowhere to migrate as their optimal climate zones shift upward beyond the mountain peaks. Understanding island biogeography and the processes that drive evolution in isolation provides crucial insights for conservation biology. The principles discovered through island studies apply to the increasingly fragmented landscapes created by human activities, where populations become isolated in habitat patches surrounded by unsuitable terrain. As we explore the connections that link these isolated communities across landscapes, we begin to see how maintaining corridors and connectivity becomes essential for preserving biodiversity in our fragmented world. Life does not respect the boundaries that humans draw on maps. Animals migrate across continents, following ancient pathways encoded in their genes. Seeds travel on wind currents that carry them hundreds of miles from their parent plants. Pollen drifts between isolated populations, maintaining genetic connections across fragmented landscapes. Rivers serve as highways for aquatic organisms while simultaneously creating barriers for terrestrial species. These movements of organisms, genes, and propagules create the flowing networks that link isolated habitats into functioning landscape scale ecosystems. The concept of connectivity reveals how biodiversity persists not just within individual habitats, but through the dynamic exchanges that occur among different locations across space and time. No ecosystem exists in complete isolation. Even the most remote island receives occasional visitors carried by ocean currents or blown by storm winds. The most isolated mountain peak experiences genetic exchange when rare, long distance dispersal events connect it to populations hundreds of miles away. These connections, however tenuous they might seem, often prove crucial for maintaining the genetic diversity and population stability that allow species to persist over evolutionary timescales. Migration represents one of the most spectacular forms of biological connectivity, linking ecosystems across vast distances through the annual movements of countless organisms. The Arctic tern completes perhaps the most extraordinary migration of any animal, traveling roughly 44,000 miles annually as it follows summer between the Arctic and Antarctic. This epic journey connects marine ecosystems across both hemispheres as the terns feed in productive polar waters during each hemisphere's summer season. But migration extends far beyond these flagship species to encompass movements that connect virtually every type of ecosystem. Monarch butterflies link the temperate forests of Canada with the mountain forests of central Mexico, traveling up to 3,000 miles to reach overwintering sites that no individual butterfly has seen before. Caribou traverse the vast expanses of Arctic tundra, their movements distributing nutrients across the landscape while connecting different seasonal habitats essential for their survival. These migrations create pulsed connections between distant ecosystems, transferring energy and nutrients in ways that can dramatically influence the communities at both ends of the journey. When millions of sockeye salmon return to spawn in Alaskan streams, they bring marine-derived nutrients deep into terrestrial ecosystems. Bears, eagles, and other predators consume the salmon, while the decomposing carcasses of spawned-out fish fertilize riparian forests. Trees near salmon streams grow faster than those in comparable areas without salmon runs, demonstrating how migration can influence ecosystem productivity across the landscape. Seasonal migrations also create temporal connections that link the same locations across different times of year. Many bird species that breed in northern temperate forests migrate to tropical regions during winter, connecting these ecosystems through their annual cycles. The timing of these migrations has evolved to match resource availability at both ends of the journey, with birds departing breeding areas as food becomes scarce and arriving at wintering grounds when resources are most abundant. Seed dispersal creates another fundamental type of connectivity, determining how plant species colonize new habitats and maintain genetic exchange among populations. Different dispersal mechanisms create different patterns of connectivity across landscapes. Wind dispersed seeds can travel enormous distances but settle in unpredictable locations, while animal dispersed seeds follow the movement patterns of their dispersal agents, creating more directed connections between suitable habitats. Large mammals serve as long-distance seed dispersers, carrying fruits and seeds across landscapes as they forage and travel between different areas. Elephants can disperse seeds tens of kilometers from parent trees, maintaining genetic connections among forest patches separated by savanna or human-modified landscapes. Fruit bats in tropical regions regularly travel between isolated forest fragments, pollinating flowers and dispersing seeds that maintain plant diversity across fragmented landscapes. Birds create particularly important dispersal connections because of their mobility and broad habitat use. Many fruit eating birds regularly move between forest patches, carrying seeds in their digestive systems and depositing them hours or days later in distant locations. These movements can maintain plant populations in small habitat fragments that would otherwise lose species through local extinctions. River systems create linear corridors that facilitate movement for aquatic organisms while serving as barriers for many terrestrial species. Fish populations in different parts of a watershed remain connected through the flowing water that links tributaries to main stems and eventually to the ocean. This connectivity allows recolonization of stream reaches that experience local extinctions due to drought, pollution, or other disturbances. However, human modifications to river systems increasingly disrupt these natural connections. Dams fragment river ecosystems, preventing fish migrations and isolating populations that were previously connected. The Columbia River system in the Pacific Northwest once supported massive salmon runs that connected the ocean to tributaries hundreds of miles inland. Today, a series of dams has eliminated salmon from much of the upper watershed, severing connections that existed for thousands of years. Riparian corridors along rivers and streams serve dual functions, providing habitat for many species while creating linear connections that facilitate movement across otherwise fragmented landscapes. These green corridors often support higher species diversity than the surrounding matrix, particularly in agricultural or developed landscapes where natural habitat is scarce. Many animals use riparian corridors as movement pathways, traveling along streams and rivers to access different habitats or escape unfavorable conditions. Landscape corridors designed and managed specifically to enhance connectivity have become important tools for biodiversity conservation in fragmented environments. These corridors can take many forms, from narrow strips of natural vegetation connecting larger habitat patches to broad swaths of restored habitat that allow natural movement patterns to persist. The effectiveness of corridors depends on their width, length, habitat quality, and the specific needs of the species they are designed to serve. Research has demonstrated that even relatively narrow corridors can significantly enhance connectivity for many species. Butterflies, small mammals, and birds readily use corridor connections between forest fragments, increasing gene flow and reducing extinction risk in isolated populations. However, corridor effectiveness varies among species, with some organisms requiring wider corridors or specific habitat features within the corridors to facilitate movement. Mountain ranges create natural corridors that allow species to track changing climatic conditions by moving to different elevations. As climate warms, species can maintain suitable temperature conditions by moving upslope, effectively migrating vertically rather than horizontally. These elevational migrations require continuous habitat connections from lower to higher elevations, making the protection of intact mountain ecosystems crucial for climate change adaptation. The Rocky Mountain Cordillera exemplifies this type of connectivity, providing continuous habitat gradients that allow species to move across vast elevational ranges. Many plant and animal species have distributions that span thousands of feet in elevation, using different zones during different seasons or life stages. Climate change is already causing detectable shifts in species distributions along these elevational gradients, with many species moving upslope as temperatures warm. Gene flow represents perhaps the most fundamental form of connectivity, maintaining genetic diversity within species and reducing the risk of inbreeding in small populations. Even occasional movement of individuals between populations can have disproportionately large effects on genetic diversity, preventing the genetic deterioration that often occurs in isolated populations. For many species, maintaining genetic connectivity requires preserving movement pathways that may be used infrequently but remain essential for long-term population viability. Fragmentation disrupts natural connectivity patterns, creating isolated habitat patches where species may persist for some time but gradually lose genetic diversity and become more vulnerable to extinction. The process of habitat fragmentation occurs at multiple scales, from the subdivision of large continuous habitats into smaller patches to the creation of barriers that impede movement between existing habitat areas. Edge effects complicate the impacts of fragmentation by altering conditions within habitat fragments. The boundaries between natural habitats and human-modified landscapes create distinct microclimates that can extend hundreds of meters into habitat fragments. These edge environments often support different species assemblages than interior habitats, effectively reducing the amount of suitable habitat for species that require interior conditions. Climate change adds urgency to maintaining landscape connectivity, as many species will need to track shifting climate conditions by moving to new locations. Historical climate changes occurred slowly enough that most species could adapt through gradual range shifts. Contemporary climate change is occurring much more rapidly, requiring more frequent and longer distance movements to track suitable conditions. Species with limited dispersal abilities face particular challenges in adapting to rapid climate change. Plants that depend on wind or gravity for seed dispersal may be unable to migrate fast enough to track suitable climate conditions. Similarly, animals with small home ranges or limited mobility may find themselves stranded in areas that no longer provide suitable habitat. Conservation strategies increasingly emphasize maintaining and restoring connectivity to help species adapt to environmental change. Large-scale corridor networks that span climatic gradients can provide pathways for climate-driven range shifts. Stepping stone habitats that provide temporary refugia during long-distance movements can facilitate dispersal across otherwise inhospitable landscapes. As we turn our attention to humanity's complex relationship with biodiversity, we begin to see how our species has become both a major force disrupting natural connectivity and a potential steward capable of restoring and maintaining the connections that sustain life's diversity across our shared planet. Among all the species that call Earth home, one has achieved something unprecedented in the planet's long history: the ability to consciously reshape entire ecosystems and influence the evolutionary trajectory of countless other organisms. Humans have become a geological force comparable to volcanoes and glaciers, capable of altering atmospheric chemistry, redirecting rivers, and creating new habitats while simultaneously destroying others. Yet we remain utterly dependent on the intricate web of life that surrounds us, relying on biodiversity for everything from the air we breathe to the medicines that heal us. This paradox defines humanity's relationship with biodiversity. We are simultaneously the greatest threat to life's diversity and its most capable protector. Our species has driven countless others to extinction, yet we are also the only species capable of deliberately conserving threatened organisms and restoring damaged ecosystems. Understanding this complex relationship requires examining both our destructive impacts and our potential for stewardship, recognizing that the future of biodiversity largely depends on the choices we make in the coming decades. The human impact on biodiversity began long before the Industrial Revolution, though its scale and intensity have accelerated dramatically in recent centuries. Archaeological evidence reveals that the arrival of humans on previously uninhabited continents often coincided with waves of extinctions, particularly among large mammals. Australia lost over 90% of its large animal species within a few thousand years of human arrival. North and South America experienced similar megafaunal extinctions following human colonization around 15,000 years ago. These early impacts resulted from a combination of direct hunting pressure and habitat modification through the use of fire. Indigenous peoples around the world developed sophisticated fire management practices that created mosaic landscapes of different vegetation types. While these practices often enhanced biodiversity by maintaining habitat diversity, they also represented the beginning of large-scale ecosystem management by humans. The development of agriculture marked a fundamental shift in humanity's relationship with nature as we began deliberately selecting and modifying other species to serve our needs. The domestication of plants and animals created entirely new evolutionary pressures, leading to the development of crop varieties and livestock breeds that could not survive without human care. This process of artificial selection demonstrated our species' power to direct evolutionary change, though initially on a relatively small scale. Agricultural expansion required the conversion of natural habitats to farmland, beginning a process of habitat loss that continues today. The development of permanent settlements and growing human populations increased pressure on surrounding ecosystems as people harvested wild resources for food, fuel, and materials. However, traditional agricultural systems often maintained significant biodiversity, particularly compared to modern industrial agriculture. Many indigenous cultures developed agricultural practices that worked with, rather than against, natural ecological processes. Polyculture systems that grew multiple crop species together often supported higher biodiversity than surrounding natural habitats. Traditional agroforestry practices created complex, multi-layered systems that provided food, fiber, and medicine while maintaining forest cover and supporting wildlife populations. The Industrial Revolution accelerated human impacts on biodiversity through rapid population growth, urbanization, and the development of technologies that allowed more intensive resource extraction. The burning of fossil fuels began altering atmospheric chemistry, while industrial processes released novel chemicals into the environment. Transportation networks facilitated the spread of invasive species to new continents and islands, disrupting ecosystems that had evolved in isolation. Modern agriculture has become increasingly simplified and industrialized, replacing diverse natural ecosystems with monocultures that support few species other than the crops themselves. The widespread use of pesticides and herbicides has reduced populations of non-target species, while the mechanization of farming has eliminated many of the habitat features that once supported wildlife in agricultural landscapes. Urban development represents another major driver of biodiversity loss, as cities and suburbs replace natural habitats with built environments that support only a subset of native species. However, urban areas also create novel ecosystems that can support surprising biodiversity particularly when designed with natural processes in mind. Urban parks, green roofs, and restored wetlands within cities demonstrate how human designed landscapes can provide habitat for many native species. Climate change, driven primarily by human emissions of greenhouse gases, poses perhaps the greatest long term threat to global biodiversity. Rising temperatures are shifting the geographic ranges of species faster than many can adapt or migrate. Ocean acidification threatens marine ecosystems, particularly coral reefs and other organisms that build calcium carbonate structures. Changing precipitation patterns are altering the water cycles that many ecosystems depend on. The rate of contemporary climate change exceeds anything experienced during human evolutionary history, creating novel challenges for both human societies and natural ecosystems. Many species lack the dispersal ability or genetic diversity needed to adapt to rapidly changing conditions. Ecosystems that have remained stable for thousands of years may shift to entirely different states within decades. Despite these sobering impacts, humans have also developed unprecedented capabilities for understanding and protecting biodiversity. Scientific research has revealed the intricate connections that link species within ecosystems and the essential services that biodiversity provides to human societies. This knowledge forms the foundation for conservation strategies that can effectively protect threatened species and restore damaged ecosystems. Protected areas represent one of humanity's most successful biodiversity conservation tools. National parks, wildlife refuges, and other protected areas preserve habitats and provide refugia where species can persist despite pressures in surrounding landscapes. The global network of protected areas now covers over 15% of Earth's land surface, though coverage remains uneven and many ecosystems remain underrepresented. The effectiveness of protected areas depends on their size, connectivity, and management quality. Large protected areas can maintain ecological processes that smaller reserves cannot support, while connectivity among protected areas allows species to move between different habitats and maintains genetic exchange among populations. Effective management requires understanding the ecological needs of the species and ecosystems being protected. Restoration ecology has emerged as a powerful tool for repairing damaged ecosystems and recreating habitats where they have been lost. Successful restoration projects have reestablished prairies on former agricultural land, restored wetlands in drained landscapes, and reforested degraded watersheds. These efforts demonstrate that human intervention can reverse some of the damage we have caused to natural systems. However, restoration remains challenging and expensive, particularly for complex ecosystems with many interacting species. Restored ecosystems rarely achieve the full diversity and functionality of intact natural systems, emphasizing the importance of protecting existing habitats rather than relying solely on restoration to maintain biodiversity. Conservation biology has evolved from a focus on individual species to ecosystem based approaches that consider entire landscapes and the connections among different habitats. This shift recognizes that species cannot be preserved in isolation, but require the ecological context that supports their populations and evolutionary processes. Modern conservation strategies increasingly emphasize working with local communities and indigenous peoples who often possess detailed knowledge of local ecosystems and traditional practices for sustainable resource use. Community based conservation programs can be more effective and equitable than top down approaches that exclude local people from decision making about their natural resources. Technological innovations are creating new tools for biodiversity conservation, from satellite monitoring of deforestation to genetic techniques for identifying cryptic species and assessing population health. Environmental DNA sampling allows researchers to detect rare or elusive species from water or soil samples, while camera traps provide unprecedented insights into animal behavior and population dynamics. Captive breeding programs have successfully prevented the extinction of numerous species, though reintroducing captive bred animals to the wild remains challenging. Seed banks preserve genetic diversity of plant species, providing insurance against extinction while maintaining material for restoration projects. These ex situ conservation methods complement but cannot replace the protection of species in their natural habitats. The economic value of biodiversity is increasingly recognized by governments and businesses, creating financial incentives for conservation. Ecosystem service payments compensate landowners for maintaining natural habitats that provide services like carbon storage, water purification, or flood control. Ecotourism generates revenue that can support conservation while providing economic alternatives to destructive resource extraction. However, economic approaches to conservation raise questions about reducing nature's value to monetary terms and the potential for market failures to undervalue biodiversity. Many ecosystem services remain difficult to quantify economically, while the intrinsic value of species and ecosystems cannot be captured in financial calculations. As we look toward the future, the relationship between humans and biodiversity will likely depend on our ability to develop sustainable ways of meeting human needs while maintaining the ecological processes that support all life. This challenge requires not just technological solutions, but fundamental changes in how we perceive our relationship with the natural world and our responsibilities as the dominant species on Earth. As dawn breaks over a world transformed by human presence, we stand at a crossroads that will determine the biological legacy we leave for future generations. The choices we make in the coming decades will shape the composition of Earth's living communities for millennia to come. We possess both the knowledge and the tools necessary to protect and restore much of the biodiversity that remains. Yet we also face unprecedented challenges that require fundamental changes in how we organize our societies and economies. The future of biodiversity will not be determined by a single solution or approach, but by the integration of multiple strategies that address the complex, interconnected causes of biodiversity loss. This requires moving beyond the traditional boundaries that separate conservation from other human activities, recognizing that sustaining biodiversity demands transforming how we produce food, generate energy, design cities, and organize economies. Climate change adaptation will become increasingly central to biodiversity conservation as the pace of environmental change accelerates. Traditional conservation approaches that focused on protecting static reserves will need to evolve toward dynamic strategies that facilitate species movement and ecosystem adaptation. This includes creating corridor networks that allow species to track changing climate conditions, protecting climate refugia, where species can persist during periods of rapid change, and actively assisting the migration of species that cannot move quickly enough on their own. Assisted migration, the deliberate movement of species to new locations where they can survive under changing climate conditions, represents a controversial but potentially necessary tool for preventing extinctions. This approach requires careful consideration of ecological risks, as introducing species to new areas could create invasive species problems or disrupt existing ecosystems. However, for species with limited dispersal ability facing rapid habitat loss, assisted migration may provide the only alternative to extinction. The concept of novel ecosystems acknowledges that many areas will develop new combinations of species and environmental conditions that have no historical precedent. Rather than trying to restore these areas to some previous state, management may need to focus on maintaining ecosystem function and maximizing biodiversity within these new ecological configurations. This approach requires developing new metrics for success that emphasize ecological processes and services rather than strict adherence to historical species compositions. Restoration ecology will play an increasingly important role in expanding the amount of habitat available for biodiversity conservation. Large-scale restoration projects have demonstrated the potential for recreating functional ecosystems across substantial landscapes. The restoration of the Atlantic forest in Brazil, efforts to restore prairie ecosystems across the Great Plains, and projects to rebuild coral reefs show how human intervention can reverse some of the damage caused by past activities. However, successful restoration requires long-term commitment and adaptive management that responds to changing conditions and new understanding. Restoration projects must be designed not just to recreate historical conditions, but to create resilient ecosystems that can adapt to future environmental changes. This may involve using genetic material from multiple source populations, introducing functional diversity that provides insurance against future stresses, and designing restored landscapes that facilitate connectivity and species movement. Urban ecology presents both challenges and opportunities for biodiversity conservation as an increasing proportion of humans live in cities. Urban areas can be designed to support significant biodiversity through green infrastructure that provides habitat while delivering ecosystem services to urban residents. Urban forests, constructed wetlands, green roofs, and wildlife corridors can create networks of habitat that support native species while improving air and water quality, reducing urban heat islands, and providing recreational opportunities. The integration of biodiversity considerations into urban planning requires new approaches to city design that view urban areas as ecosystems rather than simply built environments. This includes designing buildings and infrastructure that accommodate wildlife, creating urban agricultural systems that support biodiversity, and managing urban waterways to provide habitat while controlling flooding and improving water quality. Agricultural transformation represents perhaps the greatest opportunity for biodiversity conservation, given that agriculture currently occupies about 40% of Earth's land surface. Sustainable agricultural practices that integrate biodiversity conservation with food production can support both human food security and ecosystem health. Agroecological approaches that mimic natural ecosystem processes can maintain soil health, control pests naturally, and provide habitat for wildlife while producing food. Diversified farming systems that grow multiple crops and integrate livestock with crop production can support much higher biodiversity than simplified monocultures. Traditional polyculture systems demonstrate how agricultural landscapes can maintain significant biodiversity while producing diverse foods and other products. Modern applications of these principles include silvopasture systems that integrate trees with livestock grazing, agricultural systems that incorporate native plants for pest control and pollination services, and farming practices that maintain habitat corridors connecting different areas of the landscape. The development of cellular agriculture and other alternative protein sources could reduce pressure on natural ecosystems by decreasing the land area needed for livestock production. Cultured meat, plant based proteins, and insect farming require much less land, water, and energy than conventional animal agriculture while producing fewer greenhouse gas emissions. These technologies could free up vast areas of land for ecosystem restoration while reducing one of the major drivers of biodiversity loss. Marine protected areas and sustainable fisheries management will become increasingly important as ocean ecosystems face mounting pressures from climate change, pollution, and overexploitation. Large marine protected areas that encompass entire ecosystems can maintain ecological processes that smaller reserves cannot support. The designation of marine protected areas in international waters represents a new frontier for ocean conservation that requires unprecedented international cooperation. Sustainable fisheries management that considers ecosystem impacts rather than focusing solely on individual species can help maintain the complex food webs that support marine biodiversity. This includes protecting forage fish that serve as food for larger predators, managing fishing impacts on seafloor habitats, and reducing bycatch of non-target species. Aquaculture can potentially reduce pressure on wild fish populations, though it must be developed in ways that minimize environmental impacts and avoid introducing diseases or invasive species to natural ecosystems. Technology will provide increasingly sophisticated tools for monitoring biodiversity and managing conservation efforts. Satellite imagery and remote sensing can track deforestation, habitat degradation, and species movements across large landscapes. Environmental DNA sampling allows detection of rare species and monitoring of biodiversity in areas that are difficult to survey using traditional methods. Artificial intelligence can process vast amounts of data from camera traps, acoustic monitoring devices, and citizen science observations to track changes in species populations and distributions. However, technology alone cannot solve the biodiversity crisis. Conservation success ultimately depends on addressing the underlying social, economic, and political factors that drive biodiversity loss. This requires creating economic systems that value natural capital alongside financial capital, developing governance systems that can manage resources sustainably across multiple scales, and fostering cultural values that recognize the intrinsic worth of biodiversity. International cooperation will become increasingly important as biodiversity conservation challenges transcend national boundaries. Migratory species require coordinated management across their entire ranges, while climate change creates the need for landscape scale conservation planning that spans multiple countries. The development of global biodiversity governance frameworks that can effectively coordinate conservation efforts while respecting national sovereignty represents one of the major institutional challenges of the 21st century. Indigenous peoples and local communities will play crucial roles in future biodiversity conservation as they manage or have tenure rights over many of the world's most biodiverse areas. Supporting indigenous land rights and traditional management practices can be one of the most effective approaches to biodiversity conservation while also addressing social justice concerns. Indigenous territories often have lower deforestation rates and higher biodiversity than other land management regimes, demonstrating the effectiveness of traditional conservation approaches. Education and public engagement remain fundamental to conservation success, as sustainable biodiversity conservation ultimately depends on widespread public support and individual behavior change. This includes not just formal education about ecology and conservation biology, but also experiential connections to nature that foster emotional connections to biodiversity. Urban nature experiences, citizen science programs, and nature based recreation can help maintain human connections to the natural world, even as more people live in cities. The economic transformation needed to sustain biodiversity will require developing new models that account for the true costs of environmental damage and the benefits of ecosystem services. This includes removing subsidies that encourage environmentally destructive activities, implementing payment systems for ecosystem services, and developing green finance mechanisms that direct investment towards sustainable practices. As we look toward the future, the challenge of sustaining biodiversity in an era of rapid environmental change will require unprecedented cooperation, innovation, and commitment. Yet the scientific understanding, technological tools, and growing awareness of biodiversity's importance provide reasons for hope. The same human ingenuity and collaborative capacity that created our current environmental challenges can be directed towards solutions that sustain both human societies and the magnificent diversity of life that shares our planet. The future symphony of life on Earth will be composed by the choices we make today. We have the opportunity to ensure that this symphony continues to include the voices of countless species, each contributing their unique notes to the grand chorus that has been playing for billions of years. The harmony we create will echo through generations, a testament to our understanding that human well being and the well being of all life are inextricably intertwined.